Well, welcome everyone. I'm Lee Faylow and I'm chair of the executive committee of the Sydney Dementia Network. And I'd first like to start by acknowledging that we are on the unceded lands of the Gadigal people of the Eora Nation and acknowledge elders past, present and emerging, and welcome any First Nations people to our meeting today. Welcome to all of you. So this is not the formal conference. Those of you who've never been to an AGM, this is our annual general meeting where essentially once a year we report back on what the committee's been doing and get input on what the committee should do in the next year. So those of you online, welcome. This is the AGM and I don't think we can take questions from you. So if you just want to go for lunch and come back at 1, you're also very welcome. Right. So for those of you who don't know, the Sydney Dementia Network is a loose network coordinated thing that's sponsored initially by the DVCR of the University of Sydney, but now supported by Brain and Mind Centre. And basically, we're a group of researcher clinicians, and our remit is to support research and research translation into practice in the greater Sydney region. And we've been going for 6 years, be 7 years, who can remember? 6 years, about that. So we have a very gripping agenda, and it looks very similar to what AGM agendas looks like, where we'll get approval for the minutes. I'll overview what we've spent our budget on, and then we'll talk about the things we've been doing and get input on what we might do in the coming year. So BMC support the activity of the Sydney Dementia Network to a budget of $20,000 a year. And so this is what we spent our money on last year, $9,000 on this symposium last year. We spent $7,000 giving future research leader awards, which were essentially travel scholarships. We ran another workshop, which we spent $900 on, and we spent $800 on vouchers to support the Lived Expert Experience Advisory Panel. That's it. And we hope that the Brain and Mind Centre will support us. Well, we have $20,000 for this year, 2025, and we hope that we will get $20,000 next year as well. So here are some of the committee members. I'm going to read the list because if not, I forget people, and then that's embarrassing for me. So let's just wave as I call you. So the other people on the committee are Olivier Pigray, who's there, Moira Irish, who's hiding, she's over there, Carol Dobson-Stone, whose hair looks fabulous today, Rosanna Porto, Rosa Porto, Penny, Penny's inside now, Penny Monroe, Johannes Mikalian, and Jackie Wesson. So all these wonderful people volunteer their time, and we're very ably supported by Shannon Camilleri, who's our secretariat. And we have meetings. I think they're productive meetings because in those meetings we do things, and I'll show you some of the things that we do. We don't just have meetings to chat, though there is a bit of chatting sometimes. So we put out a newsletter. Well, Shannon's got the newsletter that we feed it into. We ran a conference, 142 people with 65 in person last year. Hopefully same numbers this year, Shannon. Yeah. And we have 433 members, which loosely just means those are people on the email list. So last year at the AGM, some non-University of Sydney people suggested we do more cross-university collaboration. So we did have Rosanna join the committee. We were all University of Sydney people before, which I think has been great. So we've had more to do with then Western Sydney University, and we've got more speakers from other universities in Sydney at this year's symposium as well. Dimity Pond, a GP, suggested that we engage with GPs. So I did reach out to the University of Sydney GP Clinical School, which is a really interesting meeting because none of the GPs there were really interested in dementia research. We had a very delightful conversation, and then they told us about like their clinical experience of dementia, but there was no, I guess, further conversation around incorporating training around dementia in the GP training or anything like that. I tried though. I can see Jane Thompson shaking her head. I did try to engage with the GP clinical school here, and we've been doing other work with general practice in Western Sydney and Southwestern Sydney, and it's really, really difficult. It's clear to me that GPs don't find dementia very high priority. So if we really want to engage with them, it's a big push. The other thing that the broader you suggested last year is that it is useful to have award schemes to recognize EMCR research. So we have the Future Research Leaders Award, which is unfortunately only open to University of Sydney researchers, but we also have a Research Impact Award, which is open to anyone. So you can put that on your CV, and we thought that that would be good practice for people to write their research impact statements for grants, essentially. You might be interested to know that we wrote, the committee wrote a rubric around research impact, and we actually did quite a lot of discussion and not quite arguments, but there were quite heated discussions about what research impact should be and how you document it, particularly for basic science or someone really early in the career, and how, I guess, difficult it is. And I think the big thing I learned from that, if I were EMCR, is that you need to have thought about impact and have an impact kind of plan ready, even if you haven't actually got the actual impact. Does that make sense? You've talked to the stakeholders, you kind of know who you want to impact, and that's kind of clearly documented, and you've started having those conversations. We have also engaged more broadly with the local health districts in our region. Most, I guess, with Sydney Local Health District, I actually spoke at their mental health conference in Concord earlier this year, at the end of last year, I can't remember. But yeah, we talked with the geriatrics CAG at Northern Sydney Local Health District, particularly, and we've been also talking with Western Sydney LHD, Southwestern Sydney LHD. We've also facilitated researchers from the network to give public talks, Inner West Council, Canada Bay Council, other councils as well. And we continue to chat or engage with the New South Wales Agency for Clinical Innovation and ACI Age Health Collaboration Network. So we get lots of emails. Well, I say we, Shannon gets lots of emails into the Sydney Dementia Network email box. And usually we try and figure out who the right person for them to talk to is, and kind of like suggest researchers that they could work with and stuff. Oh, Shannon's gone. Bye, Shannon. So I'm happy to announce that we will run our Future Research Leader Award again this year. We have 2 awards of $1,000 each for national travel, and 2 awards of $2,500 each for international travel. You must be a staff member or formally affiliated with the University of Sydney and presenting research related to dementia, and hold a PhD but be less than 10 years postdoc or equivalent. And the formal call will open soon, and if you're getting the newsletter, you'll get that newsletter, but we've already decided this basically, right? Yes, Shannon's not here to nod at me. Thank you for those of you who applied for the Sydney Dementia Network Research Impact Awards as well. And I'm happy to announce the winners are Dr. Zoe Schroer. How do I say her surname? Schroer, should know. Zoe's at a wedding, not her own, and isn't here today. And to Dr. Jackie Wesson, who is on the committee but wasn't on the panel, who judged in any way and was not involved in any of the conversations. And we both invited both Jackie and Zoe to present in the symposium, and we will hear from Jackie but not Zoe today. So I think we'll probably do these again. So if you want to practice writing your impact statements, please do. EMCRs, who's speaking on this? Jackie's.
Thanks, Li-Fei. Briefly, some of the activities that we did this year, both
successful activities. We tend to decide from feedback from EMCRs through the newsletter and so on as to what we may or may not do. So this is our second time running this one, the Dementia Australia Grant Information Session. And we partnered with Annette Moxie, who's the Dementia Australia Research Foundation General Manager. We also had Jane Thompson, who's one of our lived experience expert advisory panel members, Olivia, who is a previous staff reviewer, a couple of successful awardees, Amanda, Pradeep and Edwin. And so that was a really successful webinar, and we will likely, don't know, but it's our second year in a row, and it gets put up on the Dementia Australia Research Foundation website as well. So that's that real cross-fertilisation and strengthening and networking. And the other one was this, the basic clinical science workshop, which was held in October, aiming to bring together basic and clinical scientists in the area of dementia, just to start kick-starting ideas and get that collaboration starting early. And so we had strong ECR attendance with Paul Haber, Professor Paul Haber and Associate Professor Wendy Gold come to that. And we've also, the other thing that we do when we're talking about our profile and impact and all these sort of other metrics that we need to think about as researchers, we had a couple of research spotlights on our early career researchers in our newsletter. And so that's something that we continually do, is to really think about how to just get people's work out there. Thank you.
I was going to look at Joe. Joe, people want to be profiled in the newsletter. Can they? If you want to be if you're an EMCR, I'm just plugging it, and you want to be profiled in the newsletter so that you're somewhere.
Looking at the 10.
Yeah, please do reach out to Joe. It's a very simple process. You do need a photo that you're happy with being shared on the internet, which seems to be a big blocker for people. So get a nice headshot and then, you know, write a few easy sentences about your research for Joe and go in the newsletter. Byron.
Hi everyone, I'm Murren. I've been working on the LEAP panel for the last couple of years. So just to give a quick overview of where we're at, we founded LEAP in 2021. We have 12 active members and we're doing a call out to get some new fresh input onto the panel. And so if anyone is aware of people who have lived experience, this can be someone who's living with dementia, it can be someone who is a current or former carer of someone with dementia, please do get in touch with us. Rosanna is our new support person who's taking over from Siobhan Shaw, who was providing admin support over the last few years, and now she's finished her PhD. And so in terms of activities that we did over 2024, we've had a grant review panel where 3 researchers pitched their ideas. We've had a lot of consultation actually externally to the network. So people are starting to contact LEAP for input on various things, such as the Living with Dementia survey, various projects that are running in the uni and outside of the uni, as well as some MRFF funded projects as well. I think that's it, but I would say if you are interested in pitching your projects to get feedback, to get early advice and input on work that you're doing, please do contact us, because I think it's really invaluable and it's led to a lot of grant success over the last few years.
I'm also plugging, I guess, that if you have material that you want to go in a newsletter, please send it to the that email, the Sydney Dementia Network email. So if you're running a dementia related event online or in Sydney, we're interested in that. We're interested in dementia news, including grant success. If you're recruiting participants relevant to members, so we don't really have a lot of members, people with dementia or carers, but if you're looking for health professionals or researchers, you can certainly put something in the newsletter. Obviously, we're interested in dementia related grants, promoting them to members. And we have over 400 members now. And if you're not on the mailing list, please join. So I am sad to say that Jackie Weston, after 2 years on the, why are you sad? You're not dead, you're just leaving the committee, is stepping down from the committee. And I would like to thank Jackie for her hard work. And we have a token of I'll do a token of appreciation in a minute. Is that right? Yes, Shannon's nodding. And I would also like to say that I'm now stepping down as chair. I've been chair for 3 years, and I think you're all sick of me heading this conference. And Professor Maureen Irish is going to step up as chair this year. Thank you for taking this on. I'm very excited to see what you do. And you want to talk to this slide?
I think it's fairly.
Yeah, okay. So so these are the things that Maureen's really focused on doing. And I also wanted to, plug that because Jackie's stepping down, we now have at least one new spot. This is a very nice committee. We do have heated discussions about things like rubrics. We also did have a heated discussion about serving alcohol at the event. So that you know, it's a very nice committee, and it's good to meet other people in different bits of dementia research in Sydney and hear about what they're doing and get, it's not quite gossip, but get news about what other people are doing. So especially if you're EMCR and you want to step outside your lab or your little comfort zone, then please join. You will be expected to do some work outside those 12 committee meetings, like taking on running an event or helping, well, Rosana's helping with LEAP, but doing a little project, which you can put on your CV as something that you achieved. So I want to promote you doing that too. And now I'm going to open the floor. What? I don't have any time. How are we going for time? We got 10 minutes. Okay. Do you want to come up, Maureen? So I think, because you're the chair, so I don't want to commit to things. Chair soon. So this is the bit really when we open up the floor to you to find out, you know, what things are on your mind as dementia researchers. Are you worried because you've got NIH funding and you're being cut? Are you worried because you're a basic scientist and it's really hard to get money? Like, I can't say, we're not promising we can fix all these things, but it's really interesting, I think, to hear from you. And then if you've got very concrete, specific suggestions that you'd like the committee to do this year, remember, we have $20,000 in total and we're all volunteers, but you know, within that packet, if you can think of things you'd like us to do, we're also very open to do them. And you see that we did do some of the things that you asked for last year. It's open for discussion now. I'm not scared. Jane.
Start the ball run. I'm really pleased to see that you're going to focus a bit more on LEAP this year. We had a bit of a quiet year last year, and I think we've lost a bit of momentum. So I think working on building up LEAP and also just having maybe some clearer objectives for us and making us better known to the researchers, so researchers do come to us with their proposals. So
yeah, thanks.
Go LEAP.
Thanks, Jane. And I think Shannon is noting that. Thank you, Shannon.
I totally endorse that as well.
Thank you, Peggy. Anyone else? Carol.
Well, it was another question really, but I guess wanted to get a feel for how people would like us to run events.
Oh yeah.
Like, do you prefer online stuff? Does it enable more people to join from different places, or are you keen on more face-to-face type meetings? I guess each have their advantages.
Okay, I'm gonna say, I'm gonna ask a question that you've got to put your hands up, so you have to answer. So I'm gonna say, will you prefer online or face-to-face? You can only put your hand up once. So for those of you who would prefer online events or seminars, can you put your hand up? One. No, it's fine. No, it's no judgment. For those, yeah, no, thank you for coming from wherever you came from.
For the ones that are on.
Oh, those are you. I'm sorry, you're right. Some of you are online. We can't count the ones online. We presume that you prefer online if you're online. Shannon will count the ones online. Sorry, online people. How about people who prefer face-to-face? Okay, so all the face-to-face people prefer face-to-face.
That's very obvious.
Oh, but the online people might prefer online. Yeah, okay. What a bias sample. I should have known better to ask that question. Okay, any other thoughts about things you would like us to do for events, for talks.
Well, I guess we did a bit of outreach this year to help promote EMCRs to talk to the general public. So is that something that people would be open to doing more of next year?
Yeah, if you want to give a public talk, tell us, I think. It will be, if you want to give a public talk, it will usually be on the topic that the people asking for the talk want, which are typically about healthy aging or a lot preventing dementia, maybe what is dementia. Sometimes they ask about testing, like
Living
stuff,
well.
living well with dementia. So if you want to give a public talk and you can pitch your talk in a way that's accessible for your mum, okay? You can't give your normal research talk because it's bamboozling if you're not a researcher. Then please let us know, because we do get asked to give talks. And sometimes I get asked to give talks and I can't give them. And we're happy to then give people opportunities to give talks. And it's really nice for your CV. And it's a really nice way of meeting the public and knowing what they're worried about and getting out of our ivory tower. Yeah. Rosanna.
I think it's something that we have been talking in the meetings, which is bringing the basic science a little bit more.
Yeah.
But I think we're still struggling to reach out to basic science, because when we did the network, we didn't have much of basic science as well. And I think that's something that we can try to work on. How is our reach to basic science?
I don't know. Should we be running something? I don't know. There's a few basic scientists. We have 2 basic scientists on the committee. I'm not looking at you to give you a job. Like both Carol and Rosanna are on the committee representing basic science. And we did try and run something for basic scientists. It was okay, but it wasn't like as successful as we would have liked. And we did have a conversation a couple of years ago around basic science advocacy with Carol in particular has been kind of working on that. We've been like, we need to like help basic scientists be competitive, I guess, in the current funding environment.
Can I maybe chime in? And
yes,
I'm a basic scientist, to those of you who don't know me. So I guess one of the things from a basic science perspective that is also important, we like engaging with clinical research, but we really still are inherently basic science. And so I guess if there are ways to still maintain, so we don't want to change our research questions too much, but from the advocacy angle, if there is a way, which is really hard in the current Australian environment, translation is. If there is a way from your platform that you can advocate that the foundational basic science by itself is still really important for us to understand what's causing disease and try and develop drugs, biomarkers, all of that, then maybe the basic scientists might be more keen on that angle rather than feeling all the time that we need to change our research to fit more clinically.
Yeah, that makes sense. I mean, we have talked to
building your voice.
yeah.
Because the fitting in clinically is
it doesn't quite make sense either, right?
And there's a lot of this, like we're obviously doing this in the AD team as well.
Yeah.
And there has been, from the Alzheimer's team perspective, there has been really nice engagement from the clinicians, understanding a bit more about the basic science angle, and that's been really enjoyable because the basic scientists can just be them.
Yeah. Thanks, Eleanor. So Eleanor Drummond runs the Alzheimer's disease. What's it called? The
Alzheimer's and neurodegenerative
team. At the BMC. Yes.
I always found that the Dementia Advocate program is very useful, but I think as basic scientists, we only access them like right before the grants. But I think if we are to have any program where you can actually invite the Dementia Advocates and show them around, that will actually bridge the gap so that you can direct your impact for a general audience, which is quite important here, right?
So do you think maybe some advocates would like to visit a lab or two? Maybe, yeah, okay. I think we could, 5 minutes, thank you, Shannon. I think we could definitely maybe manage a lab visit. Maybe we'll visit your lab. That would be exciting. Thank you. Yeah, that'll be excellent. Maybe I'll come and visit a lab because sometimes I find the labs confusing too. Once they start to say things like nano, I get like sick. And they show me pictures of equipment. That's also scary.
It's nice as well.
I'm okay with mice because I did mice, I did rats for my honours project, so I'm okay with the rest. Peggy.
Oh, Life, that's an excellent suggestion. I'm glad it's come up. The Walter and Eliza Hill Institute in Melbourne do exactly that with their advocates. They're very, very big on doing that. So it's an excellent idea. So
Yeah. I think we can manage it one lab visit this year. That seems achievable. Eleanor.
If I just might suggest, if you're going down that angle, which sounds amazing, philanthropy team often is doing, like, they have a whole system of bringing people in science
okay.
labs. They often tour the BMC as a whole and so them both clinical and basic science, and that seems to resonate really well with those people who are often lived experience people as well.
Yeah, just to build on Eleanor's comment that the Brain and Mind Centre will often conduct visits and tours of the appropriate labs and facilities. So if you can reach out to our team and we can organise tours together with the researchers.
Welcome, everyone. I'm going to be any other suggestions for that the network should be taking on this year? If you're too shy to say a suggestion now, you're very welcome to just find me or Moirin at the break and tell us. Otherwise, I forgot to endorse the minutes. Oh, fail as chair. Okay, to those of you who were here who had copies of the minutes, I'm looking at the executive team who were sent them. Can someone please endorse the minutes? Jackie's hands up and Olivier's, thank you, from last year. And if anyone wants a copy of the minutes, we have them if you want a copy. We thought we'd save the trees and not print lots. And hence the meeting is closed. And in a minute, we'll start the next meeting, the actual, symposium. Okay, wonderful. Yes, yes, okay. Oh, yes, sorry.
Say thank you to Jackie and say thank you to Life for their service on the Senate.
Okay, now we have to work. Okay, so you just go straight into the conference, right? Yeah. Oh, slide swap. Okay, I'm ready. Swap. Okay, we have slides. Okay, welcome to the people online. We finished the AGM and now we're starting the symposium proper. For those of you who haven't met me, I'm Li-Fei Lo, and it's my pleasure to welcome you to the 2025 SDN Symposium. I'd like to start today by saying how grateful I am. I'm grateful to have this dementia community, and I'm grateful that we've had this symposium every year for, I don't know, 6 years or 7 years, I can't remember how many years. And that we can come together in person and I can not sit at my desk for a whole day and talk to real people who care about the thing that I care about. And I learn so much and I get ideas and gossip and hear about things that are happening and catch up with old friends and meet new people. So while we do have a stellar world-class lineup of speakers, who I'm very grateful have come for the conference and prepared to speak to. It's really you that I'm really grateful for because all the chatting in the breaks are what really I think make coming to an in-person conference. Or for those of you online, don't make you feel bad. The lineup is stellar, so it's still worth being online, but really wish you were here in person. So let's really enjoy having half a day to spend together, learning, thinking, and hanging out with you. For those of you who didn't realize it, looking at the symposium program, We've loosely themed it around emerging risk factors for dementia, the ones that didn't make it onto the latest Lancet infographics. So I am expecting to be fascinated, challenged, and inspired. No pressure to our wonderful speakers, of course. But to start, I'd like to introduce Professor Kathy Rashorgi, co-director of the BMC, to do the acknowledgement. Thanks for coming, Kathy.
So I'm pretty excited about all this too, I have to say. But I just thought before we start formally and do the acknowledgement, I'd just like to correct one thing. When Lipe was saying that you had heated discussions at the committee, I'm pretty sure they were just excited discussions, right? Yeah. And I'd also like to see this little crush of, I think, EMCRs up the back sort of spread out and join us. We're okay. You're part of us. So you're very welcome to be part of the greater thing. You don't always have to sit in the back row. But I'm delighted to do this for my wonderful long-term colleague and friend, Lee Fay, and also meeting the world-class researchers like Kaarin Anstey, who actually may not remember, but I was a PhD student when you were at Neuro for a little while there. So I know a lot of you already. But we will start the formal proceedings, and it's a great pleasure, really, enthusiastically. I'd like to acknowledge the Gadi people of the Eora Nation, the traditional owners or traditional custodians of the beautiful land on which we're meeting today. And further, I'd like to pay respect to Gadigal elders past and present, and to really acknowledge the millennia-long care for country for which they were responsible. So thank you.
Thanks, Kathy.
Hi everyone, I'm Olivier Piguet. I'm chairing the first session. I've got the pleasure to chair this first session.
Hand over to the wonderful.
No, you didn't, but that's okay. Thank you, Kathy. Yeah, this first session is really exciting because we were talking during the AGM about one of the aims of the Sydney Dementia Network is to promote the interactions between basic science and clinical science. Interactions between these different aspects of research in dementia, and this first session is exactly what it is. So 3 speakers, outstanding speakers, and looking at different aspects of dementia research, one from a more clinical aspects, one more basic science, and one about some policy around Sydney dementia. So our first speaker is Kaarin Anstey. And it's my great pleasure to introduce Kaarin. I've known Kaarin for 7 years now. Kaarin trains at Sydney Uni before then moving for a very short period of time at Neuroscience Research Australia, and then went to ANU for some years, where she developed an outstanding research program looking at healthy aging and risk factors of dementia before returning to UNSW, and where she's an ARC Laureate Fellow and also the director of the UNSW Aging Futures Institute. So Kaarin's research has been focusing on dementia risk factors, risk reductions, and brain health, and has developed a number of interventions program, apps, and too many things to name, Kaarin. So it's my great pleasure to have you talk about emerging risk factors for dementia. Thank you.
Well, thank you. It's lovely to be here today. And I also wanted to acknowledge the Gadigal people, the traditional owners of the land here, and the University of Sydney, and pay my respects to elders past and present, and extend that respect to any Aboriginal and Torres Strait Islander people who are with us today. So I'm talking about emerging risk factors for dementia. So I want to start off by looking at, well, what makes something an official risk factor for dementia? Because actually all of the risk factors that were published in the Lancet Commission in 2024, there's actually been published
decades ago. And if you look back in the literature around the 2000s, so around the turn of the century, you'll see that there were publications from Eurodem, which we were talking about over lunch with a few people, publications from Professor Tony Jong looking at depression. There was work on obesity, cardiovascular risk factors, the Mediterranean diet, sleep. So when something's announced as sort of a new risk factor by the Lancet Commission or an official risk factor, what does that actually mean? And I think that's what I just want to spend a little bit of time talking about, how science progresses and we get to a consensus. And then I'm going to talk about some of the neuro research that's emerging. So just as for some of you who are from public health, this is like 101. Going back to some of the criteria that epidemiologists would use to look at what we use in causal inference to think through. But it's actually, some of this is really useful when thinking about some of the dementia risk factors, particularly sleep. So one of the real issues in establishing whether something's independently associated with an outcome is whether the cause precedes the effect. And for those of us who've been involved with dementia research, we understand now that the neuropathology underlying Alzheimer's and dementia is accumulating over decades. So it's actually very difficult sometimes to disentangle what is a result of actually the disease process. So with smoking and lung cancer, that's a clearer association between an exposure and an outcome. But some of the symptoms of dementia are actually part of the prodrome. So things like depressive symptoms, sleep dysregulation, etc. So in our area, that's quite an issue. Then there's some other criteria, like is the association consistent with existing knowledge? Have similar results been shown in other studies? That's where your meta-analysis comes in. What's the strength of the association between the cause and effect? Does increased exposure equal increased effects? So is there a dose-response relationship? And now this one about reversibility is a real issue for dementia research. So does removal of the cause decrease the risk of the effect? One of the problems we have is we're talking about risk factors for dementia, which is mostly from observational research, but we really lack trials that show the benefit of removal of that risk factor or reducing that risk factor that then show a reduction in cognitive decline. And there's almost no research showing prevention of incident dementia from those interventions. So even the finger trial, it looked at cognitive decline, well, actually less cognitive growth, if you look at the actual results from the Lancet paper. So we still, in our research, we still really need to work at that particular criteria. And then there's a couple of other criteria about the robustness of the evidence base. So this is the Lancet Commission report. So the Lancet started to publish commissions on dementia in 2017. There's been 3, and with each commission, the list of risk factors grows. And this is what people keep quoting as the risk factors for dementia, but it's actually what the Lancet Commission, the group of Lancet commissioners have agreed to by a consensus process, met sort of a criteria that they've decided on that made them the official list. It's not necessarily what the broader field, always who work in the dementia epidemiology, may agree or disagree on. And it's a very much a moving, evolving field where evidence is being published all the time. Like every day I see a new publication that's impacting this evidence base. The other thing to note is that this list of, which I won't read them all, I'll let you read them, but there's actually one really significant factor which is not on the Lancet Commission list and hasn't been on any of those 3 lists, and that is nutrition. So a lot of other people believe that dietary patterns are related to brain health. There's a huge amount of research on diet and risk of cognitive decline in incident dementia, but the Lancet Commission has never included that in its list, and it hasn't included sleep, which I know we've got a lot of people here who are very interested in. The WHO guidelines is another authoritative source on risk factors. It published guidelines in 2019, and it had a slightly different list to the Lancet Commission. In 2019, the WHO concluded that midlife cholesterol was an increased risk factor for late life incident Alzheimer's disease, but that only got accepted by the Lancet in 2024. So I'm just sort of illustrating how this is a movable picture here. So the WHO guidelines, I just spent a little bit of time talking about why their list is different from the Lancet list. The WHO guidelines focused on interventions that reduce cognitive decline. The Lancet Commission is mostly based on the observational evidence. So it's mostly looking at cohort studies where we've seen an association between an exposure and an outcome. And so when you use a different methodology, the WHO guidelines are using the grade methodology, they ask a different question. And so they've got a slightly different approach and have different risk factors. So just the general point I'd like to make is that evidence accumulates very slowly in this field. There isn't a sudden discovery often. Even if we discover a new risk factor, it has to be replicated by many groups. You have to look at adjusting for bias, etc. So, you know, we have cohort studies, you have RCTs, you have animal research. And so to get to the point where the WHO guidelines make a recommendation on a risk factor, it could be decades of research. Now, in 2019, my team conducted a very detailed evaluation of the quality of the evidence on dementia risk factors. So not just what are the risk factors, but what's the quality, you know, what countries have published data, how much replication, how much duplication is there in the systematic reviews, how recent is the evidence, you know, what's the quality of the outcome measures, etc. And from that, we found that there was systematic review level evidence on 32 risk factors for Alzheimer's disease and vascular disease, vascular dementia. Most of the data on risk factors is for all-cause dementia, and that's really just to do with the prevalence and statistical power. There's often not enough cases of vascular dementia in order to estimate risk ratios in cohort studies. In that review, we found strong evidence, and very consistent and strong evidence for depression, diabetes, high midlife BMI, low education, high homocysteine, and smoking as risk factors. And then for protective factors, we found strong evidence for non-steroidal anti-inflammatory drugs, alcohol, low levels of alcohol consumption, physical activity, fish and coffee consumption, and mixed evidence for a number of the medications. And then there was insufficient evidence for a number of other risk factors. So this is the evidence back in 2019. A lot's been published since then, but I'm just illustrating again, it's a different list again. And there's an awful lot published in systematic reviews on this topic. But this is probably more important from that paper is when we went to look at the quality of what's been published and where the data's from. So nearly all of the epidemiological data on risk factors for dementia back in 2019 was from North America and Europe. So there's a real lack of data from Africa, South America and Oceania, so our own region. We actually lack good quality epidemiology on dementia in Australia and the Pacific. And in some of the risk factors, there's almost no data on that risk factor for a whole region. And some of them are culturally influenced risk factors like diet or BMI, stress, depression, etc. So there is a need to take that into account. Now, this has now been written out about a lot. So the Lancet write about this. There's been a lot more publications in Lancet Healthy Longevity and Global Health now looking at dementia risk in different regions and estimating population attributable fraction in different regions of the world. So there's a real concerted effort to fill these evidence gaps, but it does mean that there's quite a bias in the literature. And just as an example of the bias that can come, the work on vascular risk factors published by Mia Kivipilto's group from Karolinska Institute in the early 2000s, most of those studies were actually heart disease studies based on cohorts that had very high cholesterol. They have a very different diet to us. It's a Finnish diet, and so they're very specific to a culture and a lifestyle and a time in history, and yet that's where we got a lot of the information on that basic vascular risk from. So it is important that we get our own data. So just generally in terms of emerging risk factors, the evidence is always accumulating. So you sort of get to a point where there's a consensus and we say, yes, that's a risk factor. There are very complicated domains like dietary patterns and nutrients, and they can be clarified by things like RCTs, analysis of biobanks, other things like Mendelian randomization studies. There's a lot to say about diet. I won't have time to go into it, but there's been an awful lot published on it. In fact, there's more published on dietary risk factors for dementia than any other area. And what you'll find in the literature and in things like the Lancet Commission is they're going to focus on the high prevalence risk factors because it's to inform public policy. You want to have the biggest bang for buck. You want to have the risk factors with the largest population attributable fractions, so the ones that account for the most cases of dementia, and that's a product of the prevalence and the risk ratio. So there is a sort of bias to more prevalent risk factors. So there could be something that's a risk factor for dementia that's a condition that's very, very rare. And it's just because it's not going to have a huge impact at the population level, it often doesn't get mentioned. And a real issue in the area is this reverse causation. So now I'm going to turn to some of the emerging risk factors, which is probably what everyone's more interested in the background. I wanted to start off with sleep. The Lancet Commission discussed sleep and came to the conclusion that they couldn't decide. There's a lot of problems with the length of follow-up in the sleep studies because of what I mentioned earlier about the prodrome of dementia, and it's been impossible to disentangle how much of the sleep dysregulation we're seeing is actually due to other neurodegenerative processes. There's also a lack of adjustment for things like obesity in a lot of the sleep research. So I did find a systematic review that was published last year, and the senior author is Jill Livingston, who is the lead author on the Lancet Commission. So this is from her team. She's looked at sleep, short and long sleep duration. So if you look at, and then she looked at short and long follow-up. So first of all, for short sleep duration, so less than 6 hours, she found a significant association between short sleep and incident dementia. But when you look at long follow-up, so she divided the studies into those with a short follow-up and those with a long. When you look at long follow-up, that risk ratio of 1.12, the confidence interval includes 1, so it's not significant. So the conclusion there was that the risk is only when you've got short follow-up, and so it's reverse causation, which is what she argued in the Lancet Commission. However, the same systematic review also looked at long sleep duration, 9 hours or more, and that did find a significant effect for short follow-up again, 2.2, but it also had a significant risk ratio for long follow-up of 1.74. So 74% increased risk of dementia for people who have long sleep duration. So on the basis of that, it looks like there is evidence that long sleep duration is a risk factor for dementia from this systematic review. It hasn't got a lot of studies, and we'd like to see more published, but this is the sort of work that has to be done in order to disentangle, whether or not sleep is an independent risk factor. Now, there's a lot of work looking at sleep. I'm only covering a very small proportion of it. And then there's sleep disorders. So they're, in a sense, easier to study. There is now good evidence that sleep apnea increases the risk of dementias, and this has been shown in a number of systematic reviews, and I'm just showing you one, the results from one. And so this study showed that the sleep apnea increased the risk of all-cause dementia, so 1.34 is the hazard ratio for that. It increased the risk of Alzheimer's disease and Parkinson's disease, but not vascular dementia. And this replicates other systematic reviews and other research. So there's quite a consensus now that sleep apnea is a risk factor. So hopefully we'll see that in the next Lancet Commission, and possibly if WHO guidelines, because the WHO is renewing its risk reduction guidelines, and I think sleep will be evaluated in that. Now, another really important area that I personally feel has been neglected in looking at risk of dementia is mental illness other than depression. So depression is a very well-established risk factor for dementia. There was a lot of debate about that reverse causation issue, but now we've got lots of studies from midlife showing middle-aged adults have an increased risk of late-life dementia. But there's all of the other serious mental illness which had sort of been neglected. It's kind of mentioned in the Lancet Commission in some of the discussion. So I'm just going to talk about some of the different types of mental illness. And in this first study, there was an association between anxiety and risk of dementia and psychotic disorder. There's been a lot of mixed results with anxiety, but the systematic reviews are now starting to be more consistent. So there does seem to be a sort of growing body of literature suggesting that anxiety does increase the risk of all-cause dementia. This systematic review found a risk ratio of 1.24, which means a 24% increased risk of dementia compared to people who don't have anxiety. The real problem with anxiety is comorbidity with depression. So it's really, really difficult to get studies that have been conducted where they're properly adjusted for comorbidities. So that's why I think it's very difficult to get a consensus about anxiety. There's also the issue of reverse causation with anxiety, because we do see the symptoms increase with closer to diagnosis. Now, another really important area of mental disorder is bipolar disorder. And here the evidence is quite strong that bipolar disorder is associated with an increased risk of late life dementia, and the odds ratio here was 2.96. This was, as I said, it was mentioned in the Lancet Commission, and it's been shown in things like UK Biobank, but it hasn't come into any official list. And this is where you've got a low prevalence disorder, and I tend to wonder whether it's because it's low prevalence, but it doesn't make it any less important. And I think it has very big clinical implications to know that bipolar, and particularly untreated depression, I don't know if you've seen the recent paper from the UK Biobank showing that treated depression, both talking therapy and drug therapy, is associated with reduced risk of dementia compared to untreated depression. Another area is post-traumatic stress disorder. Now, those of you who follow the literature know that there's been a number of studies by Christine Yaffe's group on veterans looking at PTSD, and there's now enough published. So it's sort of been in the literature, but it's never gotten to a consensus or systematic review. There's now enough published that I think we could say PTSD is a risk factor for late life dementia. And in this, a very nice systematic review, they looked at a number of studies, but they pulled out veterans specifically. The veterans had a 61% increased risk of dementia. They looked at the general population, and then they excluded studies with a high risk of bias, and they still found the effect. So it's very consistent. Now I'm going to move away from mental illness onto some of the other lifestyle ones that I think shouldn't be called. I mean, I don't think dietary patterns should be called emerging anymore, because I think it is a risk. It is a protective factor. But it's still not in, it wasn't included in the Lancet Commission. It was included in the WHO guidelines. The WHO guidelines makes recommendations about a healthy diet, reducing the risk of cognitive decline, and it mentions the MIND diet. Now, we looked at the MIND diet in an Australian cohort called the Path Through Life study, where we followed 1,753 participants for 12 years, and we had assessed them on the CSIRO food frequency questionnaire at baseline. And what we found was that the people who adhered more strongly to the MIND diet had a reduced risk of incident cognitive impairment and dementia. We also evaluated the Mediterranean dietary pattern in that study, and we didn't find a protective effect of the Mediterranean diet. So we only found a protective effect for the MIND diet. And there is a growing body of research. So the MIND dietary pattern in studies where they follow people to autopsies, like the Rush Memory and Aging study, they've actually found that people who adhere to MIND diet have less Alzheimer's pathology. And there's now systematic reviews of the MIND diet. This is just one. There are others that also show a consistent benefit of the MIND diet for reducing risk of cognitive decline and dementia. So just in case you're wondering, well, what is the MIND diet? It's basically a combination of the DASH, the dietary approaches to stop hypertension, and the Mediterranean diet. So it was the way the MIND diet was developed was that Martha Morris did a lot of research on the neuroprotective properties of foods, and she created a scoring system and then validated it on the Rush cohort studies. And it emphasises eating green leafy vegetables every day. So some of the other healthy dietary patterns just say you should eat so many vegetables per day. The MIND diet specifies that they have to be green leafy vegetables. They could be from any culture. So I think, you know, we've got a problem here of them being called Mediterranean and European names, but there's been work in China looking at a Chinese version using Chinese vegetables, and they were also neuroprotective. The MIND diet also emphasises eating berries at least twice a week as a type of fruit. So not just generic recommendation about eating fruit, but specifying berries or fruits. I think that probably can be expanded. That's an area where we need nutritionists to find other foods that are accessible and affordable if berries are really expensive or not accessible in some regions. And it also has low processed food. So all of the dietary literature shows that not much processed food in the healthier diets is beneficial. And it has low red meat and low dairy as well. So onto some of the other risk factors that are less spoken about. Now, I wouldn't necessarily call this emerging because I feel it's been established for a while. We include atrial fibrillation in our risk score now. It's been very strong evidence for atrial fibrillation increasing the risk of late life dementia now for some time. And there's meta-analyses that have shown this. And it also particularly increases dementia before the age of 70. So that's an emerging risk factor in the sense that it's not widely discussed when you're in lists of risk factors. I wanted to talk now about emerging environmental risks. So this is a whole new exciting area of dementia epidemiology. So we know that air pollution was included in the Lancet Commission, but researchers are now asking a lot more detailed questions like what sort of air pollution, what type of exposure, and then are there other environmental toxins or exposures that could impact brain health, cognitive decline, and incident dementia. There was one systematic review that sort of tried to look at all of the different types of environmental risk factors by Zhao et al. In 2021. And they found that solvents increase the risk of incident dementia, pesticide exposure, and they actually found aluminium, which is, I think Lee Falo will find that really funny. We've go back to the early discussions that we've had about aluminium. So that's a recent paper. I'll just go back to environmental risks. There's also research we've looked at in our Path Through Life study looking at exposure to green space, blue space, environmental complexity, closeness to transport hubs. There's all sorts of work happening, and so it is a really exciting emerging area of study. Chronic kidney disease is another area which, again, I feel it's quite well established, but it's not, when I say emerging, it's just not widely known. There's this is one example of a big systematic review that looked at EFGR and found that lower EFGR was associated with an increased risk of a range of cognitive disorders, and also even in a very sophisticated meta-regression still found the effect there. And then a number of other types of kidney disorders have been examined. But this is an example of something that could be low prevalence, and so it often doesn't get into the bigger, policy documents, etc. And the last area I wanted to talk about is infections and I don't have much on viruses, but the whole area of infections and viruses is an emerging area of risk. So this is just an example. This is a paper on sepsis that looked at exposure to sepsis in linked medical records and found a 62% increased risk of all-cause dementia in people who'd been exposed to sepsis, and there was a dose-response relationship, so severe sepsis had a stronger association. We need a lot more, this is a really general, sepsis, really general. There's studies on different viruses, different, herpes zoster. There's all sorts of things that have been published. It's quite an active area of research. So now I just want to tell you about cognitive risk. We developed a risk score so that you can go online and assess your own risk factors for dementia. This is a free risk score. We validated it on some cohort studies, and that gives you evidence-based advice on how to reduce risk, and it covers some of the main risk factors for dementia, and we've published a number of papers on that now, and we're using that in our research. And we're also conducting, we're conducting a risk reduction trial in people with subjective cognitive decline and MCI. This is funded by MRFF, and it's a randomized controlled trial looking at, it's a fully online intervention, so it's conducted remotely across Australia and has physical activity, dietary intervention, and cognitive training as the three areas that we're intervening in. So I just, in terms of finishing off on looking at population determinants, so a lot of the research up to now has been on individual risk reduction and interventions, even my own, focusing on behaviour change. The whole field now, there's a whole movement looking at population level risk. We've actually got an interest group called Platter, if anyone wants to join it. We had a meeting in New Zealand a couple of weeks ago. It's all looking at the social determinants of health, the structural determinants, and how they impact risk factors for dementia, and looking at what sorts of interventions are they economically cost effective, and what's the benefit. So, instead of just saying, oh, you've got to eat healthily, here's a mind diet, looking at the structural factors that might be preventing people from eating well and what we can do at policy level to change, things like sugar tax or having interventions around food security, etc. So that's another whole emerging area that's very active and is growing at the moment. So we do talk now about brain health equity, and that's another emerging area. It's like another whole field. So, the research started off, very much identifying the risk factors, and then with social factors, and now we're all talking about brain health equity. What can we do at a global level? What can we do at a societal level? Brain health impacts how people age. What are the big drivers of brain health equity? And we call it a winding road, starting at birth with social disadvantage and moving through the life course. So just to conclude, just summarise, obviously the evidence base is very dynamic and growing. We do have a real problem, particularly in dementia research, with this reverse causation, because we've got such a long prodrome and accumulation of neuropathology, and we have to be able to disentangle the risk factors. The effect sizes, some of the effect sizes of the social determinants and the population level determinants are quite big. They're much bigger than the effects you're seeing in drug trials. So some of these social determinants will have a really big effect if they can, and if we can intervene on them. And the frameworks that we're using now in dementia risk reduction are multi-level. So we're looking at genetics right through to societal and even historical factors, say like COVID. So it's getting much more complex. And we think prevention of dementia requires interventions at multiple levels. And that's all. And that's just acknowledge my team.
Thank you very much, Kaarin. We have time for a couple of questions. Yeah.
I know you said there's issues around prevalence, and so everything looks at all cause dementia, but I guess for people who are interested in atypical, non-Alzheimer's dementias, do you think that there's going to be differences in terms of the risk factors, or do you think it is just dementia in general?
That's such a good question. I think it is going to be a bit of both. I mean, we know things like pesticide and Parkinson's disease, so there are some really specific ones. We've seen work like from the Diane cohort, where people with high levels of physical activity have less brain atrophy over 8 years of follow-up. So, but we lack data. So, for example, risk factors for FTD, we don't have. I mean, this is where the biobanks like UK Biobank will help. We don't have big enough samples to look at the rarer types of dementia. So there's that sort of general brain health, things that are generally good for brain health, which I think will be beneficial for everything. But if there's very specific risk factors for some of the specific subtypes of dementia, that's where we're going to struggle with sample size. So my guess is a bit of both.
Thank you for your talk. It's really good. And I wanted to ask about the psychiatric disorders and the risk factor. You mentioned, I think it was depression, and non-medicated depression would be a higher risk factor. My question is about bipolar disorder, for example, when you have treatment starting when they're very young and they're treated for their whole life. Does the treatment per se, their whole life, could also be a risk factor, or is always the non-treated will be like a higher risk?
I don't think we know the answer to that yet. We'd love to know. So the best data sets are still observational, and so, but they've got a lot of risk of bias because of the factors why people are on particular medications. So yeah, we don't know. I do think that UK Biobank study is very, very impressive though, looking at untreated versus treated depression. But yeah, need more data. Yeah.
If I recall correctly, in the recent Lancet Commission, they talked about, or they commented on frailty and physical multimorbidity. Do you think that's that's something that's going to end up as a risk factor, or is it perhaps because it's too great of a thing, and that all those studies include frailty index that encompasses so many different aspects?
I find like this is, I mean, you'll get different points of view on frailty. I tend to think it's not independent. Like it's part of aging. All of these things are aging simultaneously. It's not an independent risk factor like smoking. And I mean, I think it was controversial that frailty was included in the Lancet Commission, but Ken Rockwood was one of the co-authors. So yeah, what would you expect? Yeah, that's just my personal view, but there'd be other people that think it is an independent risk factor. Yeah.
Other question? Yes.
What do you think about delirium?
Thank you for asking that. I think it is a risk. We don't include it in our risk scores. It's never in any of the literature because it's never captured in like a cohort study. I think it's a problem of the way we're studying this, that we're missing delirium, and it is a risk factor. There's a lot of evidence. Yeah. And there actually is data in MRCC fast. So we have actually been looking into whether we can somehow get a risk ratio for delirium. But yes.
David,
One more question.
one more question.
Yeah, great talk. Do you think that so these are things that bring on dementia, but do you think that in the same way they behave with those that have dementia and continue with these comorbidities, that it could accelerate their disease? Or do you think that it's more just the former than contribution of survival as well?
I think I think you're talking about sort of secondary prevention, tertiary prevention. Yeah, I mean, there is some evidence that treating sort of vascular risk factors in MCI will delay incident dementia, and that there's also some evidence that certain interventions don't have a benefit for once somebody has dementia. So it might come down to the individual intervention and risk factor. I think it's going to be quite varied. Yeah. I think MCI, SC, that's why we try to intervene in MCI and SCD, where we think there's much more benefit. Yeah.
We move on, unfortunately. Thank you. All right, so our next speaker is Anthony White. Anthony is a senior group leader for Cellular and Molecular Neurodegeneration Brain and Mental Health Program at the QIMR Berghofer Medical Research Institute in Brisbane. His research is looking at novel therapeutic approaches, and he's been looking at different disorders, motor neuron disease, and at the moment he's doing some, I find, very exciting stuff, looking at new models of neurodegeneration, looking at microglia, olfactory cells, 3D cultures, and organoids to identify new treatment potentials. And time of his talk, human monocyte-derived microglia models of Alzheimer's disease, investigating intrinsic factors and environmental effects. Welcome. Thank you.
Thank you very much for the opportunity to come down and speak here today. As mentioned, Yep. Is that better?
Yes.
Yeah. Okay. Yeah. As mentioned, we have a lot of interest in sort of environmental influences in dementia and how they, interact with the intrinsic gene changes that occur. It's really interesting coming down here. I think I was working with Simon Hawke in the UK at the Imperial College of Medicine for a number of years, and we were both coming back to Australia about the same time, and I was talking to him a lot about coming here to work when it was very at the earlier stages of setting up this institute. I didn't end up doing that. I went to Melbourne, and that was kind of because there was a more definite job offer down there, and my wife could work down there as well. So, but it was kind of a knife edge that I could end up working here from the very first time this institute was set up. So I kind of have a soft spot for this institute. I really, really like what the people are doing here. So I'll just give a little bit of background around the area of neuroinflammation, which is kind of leading into a lot of the work that we do. So as you're probably aware, neuroinflammation is really a key area that's involved in a number of neurodegenerative diseases, Alzheimer's, Parkinson's, motor neuron disease, and so on. And the key cell type that's involved in this is the microglia, which is pretty much the only specialized immune cell in the brain. And it has a lot of functions in trying to keep the brain, in its normal functional state. And really, if you don't have healthy microglia, you won't have healthy neurons. We know that microglia contribute a lot to Alzheimer's disease progression. They can release toxic factors and drive other cells like astrocytes to release their toxic factors and so on. But a lot of the work that we've done in this area has come from animal models. So what we're trying to understand is what the differences are between human and animal microglia. And there's a lot of differences around the differential expression of genes, lipids, the senescence of the cells over time and phagocytosis and so on. So particularly as the cells age and microglia age, there's a lot of changes that occur between animals and humans. So to study human microglia, there's not a lot of easy options. So you can take autopsy or biopsy material, but that doesn't give you a robust and reproducible tissue to work with. We do some work with pluripotent stem cells, but it's really hard to do large numbers of patient studies on those cells. They're quite difficult to grow. It's already if you're only doing 2 or 3 cells, but if you want to do 20 or 30, it's really quite difficult. So something that we developed over the last few years has been blood-derived microglia. So what we do is we get the peripheral blood monocytes, and we can apply interleukin-34 and GM-CSF, and we can get very nice microglia-like cells after about 2 weeks. And they express all the markers that you expect of microglia, expression of genes, and staining and so on. They uptake particles with phagocytosis, they respond to stimuli and so on. So there's a lot of similarities there with human microglia. We've done single-cell RNA-seq. We find that the 2D and 3D microglia cultures that we grow from these monocytes are very different from the original monocytes. And if we compare the gene expression to what's been published for human postmortem microglia, 2D IPS microglia, we see a very close correlation. So this is a really good but much easier model to generate microglia from patients. So microglia normally can exist in a very ramified morphology, where they're reaching out into their environment and monitoring what's happening. And then if something bad happens, they tend to shrink down to an amoeboid model where they become very reactive. So we can measure these kind of changes in microglia with the number of triple or quadruple junctions, branch length, endpoints, and so on. And we can see that when we look at things like Alzheimer's disease and mild cognitive impairment, MCI, we can see quite significant changes. We often always see more changes between AD and MCI than we do with AD and healthy controls. And so we think that that's where the major changes occurring in cells. We don't see a lot of changes in cytokines, but this is just the basal level. So we're doing more studies now on the stimulated cells and how they respond from Alzheimer's and MCI patients and so on. We do see a lot of changes in the phagocytic activity, where we see that the Alzheimer's microglia have impaired uptake of particles. And in fact, in some of the cells, you see that there's almost nothing taken up by these cells, but some other cells are perfectly normal. So it seems to be a cell-specific effect. We don't know what that means, why some cells don't take up anything and other cells are quite normal. But overall, it seems that the AD and MCI have less uptake of particles. So we did RNA-seq analysis, and a couple of the targets that we found between the the healthy controls, AD and MCI, PLRB was one of the top hits, and CHIT1. I'll talk more about PLRB in a moment, but CHIT1 is interesting because it's actually a gene that's very involved in motor neuron disease, and we're still looking at that and how it might be related to Alzheimer's. We looked at a whole range of additional cells just to confirm that these changes are there. We found that, yes, there are increases in PLRB1 expression in AD cells and also for CHIT1. And PLRB1 is actually a part of a risk gene locus that's been identified to be involved in Alzheimer's disease. It's not known what that role is, but it does occur as a risk outcome for Alzheimer's. And if we look at the staining for PLRB, we see in healthy controls, it's actually quite tight around the nucleus, and we think that it's actually occurring in the mitochondria. And then in Alzheimer's disease, quite obvious there, but you can see that it's become much more diffuse. If we put amyloid beta on the cells, we get the same kind of changes in healthy controls and even more significant changes in Alzheimer's cells. So the PLRB does seem to respond quite in microglia. But something that we're very aware of is that when you look at microglia from human or animal brains, if you take microglia out of the brain, the gene expression changes very quickly. So what the gene expression you have in the brain over 6 to 24 hours and so on, the gene expression changes very rapidly. These cells are very responsive to their environment, and a lot of those genes that are changed involved in neurodegenerative diseases. So we spent a lot of time, Carla, as part of her PhD, set up 3D cultures where she was able to grow the microglia in 3D, which we were able to identify the kind of changes that occur with over a much broader 3D environment that these cells are operating in. We found that the cells in 3D, they grew, they survived for a lot longer than they did in 2D. They really liked the 3D environment, and they had more brain-like environment compared to 2D. And we saw increased expression of the genes that are associated with microglia in these cells. We've since done single cell sequencing analysis. We found that the microglia are grown in 3D have expression of genes that indicate there are sort of subpopulations of those microglia that match what's been seen in animal and human tissues. And when we grew the microglia from people with young people to healthy controls and older people, we saw that there was increase in endpoints to the more mature microglia, more junctions, again, the more branching we see with the microglia. But the branch length was actually increased in microglia from young people, but not from older people. So that's kind of an impairment that we saw. And when we compared Alzheimer's to healthy controls, again, we saw that there were differences in points between 2D and 3D, but not in the branch length, which was just due to aging. We also saw impairments in the ramification index, so the amount of branching that you see, the number of dribble junctions and so on. So there are some impairments in the Alzheimer's cells, the microglia, compared to healthy controls. We also looked at the way microglia interact with neurons in culture. So with the green is the microglia, the red is the neurons, and the yellow is where they interact. And we were able to measure that, and we found that there was less interaction with the AD microglia with the neurons than what we saw with healthy controls and a number of contacts and so on. So they were impaired in the way that we took microglia and grew them from people with Alzheimer's. They were impaired in the way they interacted with neurons. We also added in some amyloid beta, which is the green you see here, and there's microglia interacting with that. And this is obviously the main target for the drugs that are in the clinic at the moment. We saw that the total distance that the microglia moved was higher than healthy control microglia in the velocity, so that how fast they moved was also increased. We didn't see increases in the numbers of microglia, but they were certainly more active in what they were doing. This kind of fits with studies that are showing that microglia are probably, Alzheimer's trying to remove all the amyloid material, and they are getting sort of exhaustion in terms of that process and not able to complete it. And we looked at some drugs. The dasatinib is an anti-cancer drug that has anti-inflammatory properties. It's been tested in models of Alzheimer's disease. We actually found that in our 2D model, it did substantially decrease TNF-alpha and interleukin-1 beta in Alzheimer's microglia when we treated them. But when we took the same cells from the same patients and grew them in 3D, we lost that predictive activity. So it's just that you have to be careful if you're doing studies in 2D that they may not translate into 3D. And this is just an example of what we published recently, where we can do studies on messenger RNA expression and secretion of cytokines, and try and identify patients that might have a positive response to a particular drug, whereas other patients might not have that response. We don't think that any drug is necessarily going to work on everybody, and we can identify who is the best person to put that drug into. That's something we can apply here, and we're also doing that in motor neuron systems. And we have a system at the moment where we can plug drugs into cells and look at the various outcomes if people are interested in trialing drugs on microglia from neurodegenerative diseases, as long as we can get blood for those people. So that was kind of the intrinsic work that we've been doing. So now we're tying that into the extrinsic work with looking at outside impacts. So as was just presented previously from Kaarin, something that the Lancet has mentioned that was kind of down the bottom of those slides was air pollution is seen as having a major impact on brain health and diseases like dementia. There's lots of studies in this area that are happening a lot. This has a lot of impact in brain health. And what we call landscape fire smoke. So this is anything, wildfires, bushfires, prescribed burns, any kind of smoke that's coming from anything other than sort of in the city environment. There's studies on animals, humans, and so on, showing that exposure to smoke leads to changes in cognitive function and brain, has quite significant brain impacts. So dementia is the second leading cause of death in males and the first leading cause of death in females at the moment. So anything that impacts dementia is really important. And one of the recent studies has shown that nitrogen dioxide from air pollution is the second leading modifiable risk factor for changes to the areas of the brain that are affected in dementia. So that sort of structural changes occur from these pollutants only second after diabetes and before alcohol consumption. So it's a really important area that needs to be investigated. And so while there are some studies that have been done on animal models in terms of how smoke from wildfires affects the brain, we don't really know what's happening in humans. So this is something that we've been looking at. We're very interested in looking at a number of different areas. We have a lot of collaborations setting up now to look at this across the patients in our cell models and so on. And we have a number of sources of material that we're working with. So we have filter material that you can see here. This is materials being collected on filters from the Black Summer fires compared to control filters. So there's a lot of material there that we can extract and look at. Burnt vegetation that we're looking at, and from field samples, and also generating in-house that on models. And this is just a very quick example of looking at macrophages that are being exposed to wildfire smoke extracts on filters. So this has come from field collected material from CSIRO. So you can see compared to control gene expression, this is global gene expression on macrophages exposed to material. This is eucalyptus, this is the mallee, and this is pine. So you can see there's very different gene expression with the different types of materials, how they affect macrophages that occur in the lungs and throughout the body. We're trying to understand, what this means in a lot of the studies that we see come out of the US, which is mostly around pine and so on, which is quite different to what we see from Australian vegetation. And we're really trying to understand what that means for people, and particularly frontline responders who are out there breathing this every day, and what that means for their health. And we put this material, and this was the some of the burnt vegetation material in-house. We put it onto cells from people who I think up to 30 years old and age who are over 50. And we saw that interleukin-23 was one of the really interesting cytokines that changed in response. So it was after 4 days, we saw this massive response from interleukin-23. This is secretion, not just the expression, but secretion into the media from the cells, the microglia, after exposure. And we compared it to the vehicle pollution. So we got diesel engine pollution from our collaborators and put that on cells. That actually had some really interesting effects very early on, but by 4 days it had no effect. And after 4 days, we had this very strong effect from the bushfire smoke. Extracts much stronger in age cells than young cells. So that could, has an interesting role in things like Alzheimer's disease, where these interleukins are involved, driving various pathways to drive more inflammatory outcomes. We did see also kind of similar outcomes from a number of other cytokines, not as strong as what we saw from interleukin-23, but much stronger than what we saw in younger people. So this is also important because a lot of the firefighters in Australia are volunteer firefighters. A lot of them are retired people. They're much older. In other countries, they're professional firefighters that are working in their 30s and 40s, but in Australia, a lot of them are in their 50s and 60s and so on. So it suggests that maybe they could respond differently to bushfire smoke than what we see in other countries. You can't probably see this very well, but we saw upregulation of HO1, hydroxygenase 1 exposure to bushfire smoke, particularly early on, and it's sort of stabilized. And we saw increased nuclear localization of the P65, which is an important inflammatory marker in response to the bushfire smoke. And this is very important in a number of inflammatory pathways. So yeah, just to summarize, we're trying to tie what we're seeing with the intrinsic sort of gene expression studies, and we're also working with our collaborators who are looking at polygenic risk scores and so on, how that might mean that people are more susceptible to things like air pollution and bushfire smoke and so on, and then looking at the extrinsic, how the bushfire smoke interacts with cells. And we're doing this right from the olfactory cells through to various brain cell models and organoids and so on at the moment. And trying to tie all this together. It's very early stages, but it's something that's really quite interesting. And seeing if we can come up with new therapeutic interventions. I'd just like to particularly thank Hazel and the people in my group who've been involved in this, and a lot of collaborators that have been working in this area, the funding bodies and so on. Thanks.
For question, Carol.
Hi there, thank you. That was really interesting talk. I've got some probably very simplistic questions. Hopefully you'll be able to answer very easily. But so the fact that you didn't see as much effect on various drugs in 3D models versus 2D models, is that speaking to problems with drug delivery, or is that not a problem at this kind of scale, do you think?
I think that's a problem with this scale, because the drug should be able to get. Sorry, so I don't think that's a problem at this scale, because the drug should be able to get into the cells. But there's obviously problems with drug delivery, and we're looking at those studies with blood-brain barrier models and so on. And I think it's a good question, because it is hard to compare 2D and 3D cultures in every respect. And we're actually doing a very broad study on ALS cells at the moment with drugs, and we'll probably have a better idea about how that turns out. But you know, it's a good question. But I think, you know, regardless, I guess, of what the reasons are, you just have to be careful about the way that you interpret cell culture model outcomes. And as we know, most of our models with drugs come with 2D outcomes, and we're the same. It's much easier to do, but you just maybe need to take it a step further just to make sure.
Yeah. Okay. Awesome. And with the microglia, so you were seeing some differences between young people's microglia, young people's monocyte-derived microglia and old people's monocyte-derived microglia.
Yeah.
And which is great to see, because I mean, I don't know very much about blood cell generation, but those monocytes are new monocytes, right? There's not like we only have one set of monocytes roaming around. So there's obviously some epigenetic inheritance that they have from being from an aged person that's that can
I think that was exactly, that's something that we thought was, you know, we found very interesting, that, you know, as we know that blood cells are turned over quite rapidly, and you sort of think, well, then are you going to see any differences? But when we take the monocytes and generate them into microglia, we actually do see very substantial differences between young people and older people, and disease and controls and so on. So where it's coming from, I'm not sure, but there are really strong inherent differences across all the diseases and all the age groups and so on that are reflected in when we generate the microglia, more so than if you generate iPS microglia, where you're reprogramming cells back to their early state and then generating microglia from those. We are seeing much stronger phenotypic effects with the monocyte-derived microglia, as I say, which is very interesting, that because you think, okay, they're generating new blood cells all the time, but something is being carried through in that process.
fascinating. Thank you.
I was wondering if you could talk a little bit more to why you think you saw such pronounced effects in MCI versus AD, and whether or not that reflects what we might also see in the brain and other types of cell cultures.
Yeah, initially we thought it was kind of odd, but then looking at the literature, there's a lot of cases where what you see with healthy controls is that if there's any kind of gene or proteins, if you have healthy controls in MCI, they, for example, might go down, and then in AD, they go up quite dramatically. So if you compare AD to healthy controls, you may not see a lot of difference, but you compare AD to MCI, is a big difference. And same things like, you know, they might be healthy control, MCI, they go up, and then in AD, they go down significantly. So I don't know why specifically there's large differences between MCI and AD as compared to MCI and health controls, but we saw that with our gene expression and our proteomics studies. It seems to be that's quite consistent in, at least in neofactory cell models and microglia that we've been looking at. It's quite a common occurrence, and it is out there in literature. So I think it suggests that there's sort of changes that are occurring between healthy control and MCI level, but then something is really driving it from the MCI to the AD. That's where major changes are occurring that are leading to people reaching dementia and so on. So I think that's maybe is where the interesting changes are occurring.
So a question from a non-basic scientist, and I'm probably or possibly lowering the tone of the discussion, but I'm interested in dose response related to pollution and what you were measuring. So were you measuring, and sorry, might have been very clear, but were you measuring one exposure to bushfire and then measured those cells over some days, or was it exposure over some days and then measuring the cells?
It was exposure once and then measuring it some days later. So yeah, it was interesting that when we first looked at the cells, it was kind of like over the first 24 hours, and with the bushfire smoke, we didn't see the kind of changes that we saw with diesel engines. That's very common in cities where we saw much bigger changes over the first 24 hours. And we just, I can't remember why now, but we looked for, at longer time periods, and we saw much stronger changes with the bushfire smoke after 4 days after they'd been exposed to smoke. And I'm not sure, again, I don't know why that's happening, but these cells seem to have this increase days later, which is quite different to what you see to other forms of pollution. So yeah, it's a really, really interesting question. I think we're trying to understand.
Thank you.
Can I just make a comment on that? I had the similar question, because obviously it has implications for fire people working in this environment, because obviously it's not a single exposure, so multiple exposures. So what does it mean in terms of prevention and health? Health hazards for health workers.
Yeah, actually, well, we have a project, so we have a PhD student who's working on that at the moment, and trying to understand what the exposure results in terms of working with other groups in terms of lung function changes, but also in brain health changes and so on. So yeah, there's a lot of people that we're working with the Queensland Fire Service. A lot of people are very interested in that. They're very interested in the health of the people that are working under them. We're trying to understand that. Everyone knows that it's a very complex and difficult area, and not much work has been done on it. So we'll see what we can do.
All right, unfortunately, we have to move on. So thank you. And our final speaker for the afternoon is Dr. Kim Sutherland. Oh, hello. Dr. Sutherland is or leads the Office for our OHMR, Office for Health and Medical Research, and is the acting Deputy Secretary for Clinical Innovation and Research at New South Wales Health. Before that, held director roles at the Agency for Clinical Innovations and the Bureau of Health Information. And she's talking to us about New South Wales Health Research and Innovation Strategy and relevance for the Sydney Dementia Network. Welcome.
Thank you. Afternoon, everybody. This is a complete change of pace, I think, from the previous presentations, but I'm really pleased to be here today to talk to you about our new strategy. I'd like to start by acknowledging the traditional owners, echoing the acknowledgements earlier. This was a artwork that we commissioned especially for the strategy, and really talks about how we all work together in networks and particular considerations that we need to have as we're working with Aboriginal patients in research and innovation. We also like to acknowledge and appreciate the role of consumers, patients, carers, supporters and loved ones. We know how important they are to our work, particularly at the Ministry of Health and throughout New South Wales Health in enabling system change. So I just have 10 minutes to talk to you about the strategy. So it's going to be a very whistle-stop tour. It's been over a year that we've been developing this. We've heard from over, I think by now 500 people really giving us feedback about what it is that the Ministry of Health can do and what only we can do. We understand it's a very complex ecosystem. One of the key objectives for us is to reduce duplication. So we want to be really clear around the things that we can do to enable a strengthening ecosystem and to support all the fantastic work that goes on in New South Wales and also to celebrate that work. We've done lots of surveys, workshops, really informed by the system broadly. So we're very close to publication, just going through the final political stages to be published next month. So I'm not going to go through in huge detail. This is our strategy on a page, and I'll talk to you about the main strategic directions and then reflect a little bit on how that might be relevant with a dementia lens over it. So the strategic framework is all about brilliant research, impactful innovations, excellent healthcare for all. So we have a vision statement. The strategy is based on a 10-year vision. It's a 5-year strategy, and we have a 1-year and then a series of 3-year action plans. So we're taking different time periods for doing this work. But our vision is a vibrant research and innovation ecosystem that improves the experience of delivering and receiving care, improving system efficiency and sustainability, and improving equity in access and health outcomes. That's our driving, our North Star. We, of course, want to also support New South Wales as a great place to invest, to come and do research in New South Wales. We want to beat Victoria. We've got lots of those goals. But our main goal is that vision statement. We have guiding principles that drive all of our work to be person-centred, to be clinically led, evidence-based and value-driven. And we have, I'll talk about the strategic directions, but we always have some cross-cutting themes that we really have to put a lens on any of the work that we do, and we're deliberate around that, to think about what does this mean for equity and diversity? What does this mean in terms of consumer, community and patient involvement? And how are we supporting a culture of research and innovation across all that we do in New South Wales Health? We have a number of core strengths and existing assets. We're not starting with a blank page, and we want to really be making the most of what we have already and building on those really great foundations that we have. And of course, at the base, what those foundations are our people, our partners, and our place. And so that's how we're moving forward from that great foundation. So our 6 strategic directions are, firstly, we want to create a thriving ecosystem, to adopt a coordinated, collaborative and inclusive approach to research and innovation. What we heard in a lot of the consultations was there's lots going on. There's things going on in all different areas across the system, in hospitals, in districts, in precincts, in medical research institutes. And while there is pockets and levels of collaboration, things aren't always coordinated. We aren't always really clear about how all the pieces fit together. The second strategic direction is to be strategic with our investments and to build a portfolio of investment to target areas where New South Wales is well positioned to harness current and future opportunities. This is really informed by a enhanced effort in horizon scanning. We are undertaking a regular review in order to understand where the science is very vibrant at the moment, what's going on in the science around the world, and really building on work that's done in other jurisdictions. So we can draw on PCORI, in the US, we can draw on NIHR in the UK, really looking at those horizon scanning efforts. But then what we're doing is putting it against a review of what our strengths are in New South Wales. So we're able to be informed about building a portfolio of investments of money and effort to see where we're going to have the biggest impact moving forward. Our third strategic direction is an open assets philosophy. So by that we mean developing and mobilising assets to accelerate research and fully harness emerging innovations. That means, as I say, it's a very complex ecosystem, but we do have duplication of assets. We do have things that are underutilised. We have robots in hospitals. We have a number of different pieces of infrastructure. We have different types of assets, people that we're not able to let them make the most of their opportunities. And so we are very keen to be having a drive to make sure that we are able to share assets and to get a greater return on those assets and to understand what we have available in order to drive the science and the research and innovation that we're seeking to achieve. The fourth is a place-based foundation, and that's really emerged out of a history where there was a lot of interest in building health precincts in New South Wales. And it was very much in comparison to the situation in Victoria, where there's quite a large single precinct in Parkville and is often seen as a more convenient place to come and do research, or for pharmaceutical companies or bio companies to come and work in New South Wales. Our feeling is that we're underselling the great assets of New South Wales because we do have a lot of precincts, but what we haven't done until now is join them together in a way so that it's one front door, but we're helping people to make use of the diversity and the richness of the expertise that we have here in New South Wales. So it started off with a concern to make sure we were properly using our precincts, which we had a significant period of investment in. But actually precincts aren't the only place-based foundations. We have research hubs, as I was saying, this complexity, research hubs, institutes, we have different levels of networks, and we really need to be making sense of those and making sure that we're aligning with community preferences across all those place-based foundations. Fifth is a pipeline approach, which is really thinking about, and I know you've been talking about translational research, so it is going from bench to bedside, from bedside across the system. And we've had a long history in thinking about in the health system how we get innovations, and we go through proof of concept, spread and scale, so we get into business as usual and adoption of those innovations. Historically, however, we have recognised that often we get things through a pipeline and we think they're ready to go, and then they kind of fall down and they don't quite get into business as usual. So we're really interested in thinking about how we can support getting our great innovations into business as usual, but also to support the commercialisation pipeline and to support how everyone in the community is doing that developmental work for new treatments or new models of care or new ways of supporting people in the health system. And then finally, our strategic direction, research and innovation for all. And that has really two aspects. The first is that we want all patients to be benefiting from research and innovation. Of course, it's a laudable aim, but we want the research to be translated into practice. We know that there is still significant unwarranted clinical innovation across our system. So we want to be really thinking about how we can both measure and address that. And also to increase the opportunity for our patients to be in clinical trials. We know that more and more being in a clinical trial is gold standard care for our patients, and that's something that we want to promulgate and to support across the system. It's a huge imperative for us. But research and innovation is not, we're not only thinking about it in terms of for all patients, we're also thinking about it. There's a role to play and an influence that all staff can have in this as well. And we have a pyramid of how we can both use research, we can foster research. For a few staff, they're absolutely involved in the research in conducting clinical trials, for example, or having joint appointments with universities. But there's something for all of us in research, and we should all be celebrating the great achievements of researchers in strengthening the care that we provide to our patients. And so we're really wanting to be thinking about how we can support the better use, translation, creation of research across the healthcare settings to improve outcomes, equity of access, and system efficiency. So that's it on a page. I know it's very, they can be kind of generic statements, but we feel that we should be in the generic statements for the strategy, and in the action planning, that's where we get to the much more specifics. I think I don't have much time left, but I want to just segue into very quickly the dementia action plan, because that was what you asked me to reflect on. And I think that I'm at risk of being a dilettante here, because I know that all of you have obviously very experienced in dementia, and we are in the generic enabler areas. But when I think about it, where are there overlaps, where I think there are things that seem to be resonating in this setting, but also with our strategy? The horizon scanning and prioritising effort. Of course, you can hear this is a very active area of research, how we keep up to date with what's coming out of the literature, and how we can both align our efforts in light of that, in light of those emerging areas of science, and what we need to do to enable getting on board with something that we think is really strategically important for our system. We're doing a lot of work across a lot of disease areas in diagnostics, in omics, in biomarkers. So it seems to me there's a lot of potential overlap there in the work that you're doing in the Dementia Network. There's obviously disease-modifying treatments. We're doing a lot of work with gene therapies across lots of different clinical settings. So there seems to be overlaps there. There would be opportunities to be thinking about how we could harness those opportunities together by making the most of our assets and being joined up in the system. As I say, trials are very important to us. We're really thinking about how we can make a quantum leap in clinical trials in New South Wales, and of course in dementia they're very important, and really thinking about new methods, platform trials, using new statistical methods, really drawing on expertise from across the system to help make us a very attractive place to undertake clinical trials and to support investigator-initiated trials as well. We have a long-standing program in translational research at the Office for Health and Medical Research, but the new strategy really emphasises the importance of that translational research, and especially into addressing unwarranted clinical variation. And we're very interested in alternate models of care, and by that we're thinking about how we change the who, the where, the when we provide care. How can we use AI, for example? How can we change scopes of practice so different healthcare professionals can be providing care? How can we just change things, often to provide better access in terms of in rural settings, but also they tend to be more efficient and more cost effective when we use those alternate models of care. And then finally, being into the interconnected networks in the system. Your network seems to me, I can hear the themes, very similar themes to other networks, how your network can work with other networks, and also how can we influence interjurisdictional and international conversations. So of course, we're often talking to our Commonwealth colleagues. There's currently a review of the health research strategy at the federal level. How can we be at the table and advocate for groups like yourselves, other groups, really shaping how New South Wales fits in both in the national picture, but also the international picture. So with that, I'll wrap up. Happy to take any questions, but as I say, a very different tone to all of the other presenters. So I hope you'll indulge me in the high level kind of bureaucrat speak.
2 minutes for questions.
There's one online as earlier. I saw Deborah something or other at a hand. Can't take questions online, so you've got me. I've got two. Lovely to meet you, Kim, and hope you're enjoying the role. My first question is, I didn't really hear primary care in there and community, and I'm wondering how, I imagine you anticipate connecting with primary care, because a lot of what's happening in this field is actually in the community.
Absolutely. And on a disease basis, disease by disease basis, we do engage with primary care. But of course, we're New South Wales Health, and of course, you all know who funds primary care. So there are some distinctions there, but acknowledging that in the ecosystem, of course. So we have, lots of maps in the full strategy document that's outlining how we are going to, how we do that at a more for local projects, but also how we do it as a system. And that's in the jurisdictional and the international conversations as well.
And I'm just interested in how you define or imagining translation. Because when I was working with Tony Penner, translation was often the discovery end rather than the translation end.
Well, there's research into translation of evidence, and there's translational research, and we're interested in both. And one of the other players in the system that I didn't mention was the research translation centres. So they're also, of course, an important part of the ecosystem, and we work very closely with them, making sure that we're aligning with that.
I have a very pragmatic question. Thinking of our EMCRs, maybe the ones hiding in the back row, if they're not already doing research with New South Wales Health, like how would one start having that conversation?
Yeah, well, you would apply for an EMCR grant. We have a range of those. We have grants in different focus areas, and we do think that when I said we're doing strategic investments, we really feel like the early and mid-career researchers where we should be focusing a lot of our investment, where we see there's a promising area of research. So I think we'll continue. We know that we've had great success with those EMCR grants, and we're committed to continuing those.
And would we need to be a clinician to apply for those, or can our non-clinician kind of?
Well, I don't think you need to be a clinician to apply for them. Often there's a clinical aspect to it. It depends where it is in the pipeline, but they're not all of the EMCR recipients are clinicians or working in clinical areas currently. Yeah. Thank you.
So this concludes this first session. We have afternoon tea upstairs, and we'll be back at 1:45. Yes, thank you. And a round of applause for our speaker.
So it is a great pleasure to welcome the next speaker, who is Dr. Jane Thompson. Jane is a strong and experienced advocate for public involvement in dementia research. Her background is in research, so she has an honours degree, a master's and a PhD, but her experience as a carer for her husband, who died of Alzheimer's in 2007, which led to her involvement in dementia research as an interested member of the public and a life expert. And so she has a passion to improve the experiences of those impacted by dementia, and she's been extremely valuable to us in terms of her advocacy and her sitting on our lead panel. So please give me a big welcome to Jane.
Thank you, everyone, and thank you, Kaarin. So back in about 2011, I was invited to write a chapter. Do these go down? What do you press? Yeah.
Oh, hang on.
That one.
Yeah.
Okay, so let's start again. Back in about 2011, I was invited to write a chapter for a book about the experience of carers of a person living with dementia. And it was a subject very close to my heart. My husband had died of Alzheimer's disease in 2007, and I was still coming to terms with my own experience. So I started writing. And I wrote so much that I was then offered 2 chapters in the book. But while I was doing my research, there was one statistic that really shocked me and has stuck in my mind ever since. And it was this. There was a large US study published in the New England Journal of Medicine, which examined Medicare records for more than 500,000 older couples and found that among both male and female spouses, those whose partners were hospitalised, so hospitalisation being a proxy for severe illness, during any given year had an increased risk of death. But this varied by the diagnosis of the spouse, such as that for men, the highest risk was among those whose partners were hospitalised with a diagnosis of dementia. And for women, it was highest for those whose spouse had a diagnosis of a psychiatric disease, with dementia a close second. So it was concerning that being a carer of a spouse with a diagnosis of dementia puts the caring spouse at increased risk of death. Subsequently, I came across another statistic. And this was that carers of people with dementia may have an increased risk of developing dementia themselves. Now, both of these studies, this one and the one in the New England Journal, were published some time ago, and I haven't seen them replicated nor refuted, but they're often cited. And of course, we know from many studies that being a dementia carer does have a significant impact on carers' health, but it seems that being a spousal carer might be particularly risky, and putting the carer not only at risk of death, but also of being diagnosed with dementia themselves. Now, as the theme of this conference was risk factors for dementia, I thought I would look a bit more closely at the experience of spousal carers, of course, drawing on my own experience, and why this might put the carer in increased risk of adverse health outcomes, and in particular dementia. So I have 3 dot points and a story. First, I'd just like to have a look more closely at this so-called ironic tragedy. I'd like to explore a bit more the experience of caring for an intimate partner with dementia, and then introduce the idea of public involvement as grief work. And I hope by the end of my talk, it'll be clearer what I mean by that. So, in the absence of any other supporting data, I asked Google whether spousal dementia caring is a risk factor for dementia, and the AI overview came back quite definitively using the US terminology of caregiver. It said, yes, caregivers of people with dementia have an increased risk of developing dementia themselves, and this is due to the chronic stress and isolation that caregivers experience. The longer a person is exposed to a spouse's dementia, the higher their risk of developing dementia. Older caregivers may be at greater risk for dementia because of the prolonged increase in cortisol levels and chronic inflammation. Caregivers may be more likely to experience social isolation, which can increase the risk of dementia. And they also may experience depression, anxiety, and other mental health conditions which can contribute to dementia risk. So let's look a bit more closely at the spousal caring experience and why its impact might be so profound. So to my second point. So firstly, just what proportion of carers are actually caring for a spouse or an intimate partner? And statistics from the from the UK and from Australia both indicate that about half of primary carers are actually caring for a spouse. And just as an aside, if you're looking at the images in these reports, you might be forgiven for thinking that people living with dementia and their spousal carers are all older, heterosexual, white, and that the experience is one of a doting, loving couple whose relationship is reciprocally gentle, loving, and kind. Now, these sorts of images aren't really relatable for most spousal carers, in my view. So, what are spousal carers' experiences? So, of course, some experiences will be universal and some will be unique to a particular relationship. But for me, the experience of caring for my husband was one in which I felt that if I'd been catapulted into a foreign territory where I had no roadmap and I didn't know the language. He was clearly lost, frightened, confused throughout the entire trajectory of the disease until he died less than 4 years after his diagnosis. The transitions from one level of service to the next were not seamless, and I often felt abandoned, helpless, and alone. Now, I experienced a whole gamut of emotions, which others have also reported, including intense love, loss, loneliness, anger, guilt, and of course, grief, which isn't reserved solely for the death of someone you love. It can emerge whenever there's a profound change or loss in life. Now, I've written quite a bit about this, and I presented talks, but I don't often find that people really get it. And I found it really hard to find much in publications, depictions in films, etc., that resonated with my own experience. So let me give you some insights into my experience. It's the wee hours of the morning. We've got through another day. I'm in bed beside my husband. He's in the later stages of Alzheimer's disease. We've been sleeping together for over 25 years, but now it's different. He won't get into bed unless I'm there with him. I pat him to sleep like a child. I don't sleep very well. I'm on alert in case he tries to get up and needs assistance. During the day, he shadows me around the house. If I'm out of sight, he continually calls for me. Where are you, my love? That's heart-wrenching. He wants me near him constantly, clearly fearful of being abandoned. I can't ever leave him at home alone. He's fraught with frustration and anxiety, which manifests itself physically, and he's also clearly in great emotional pain. Please help me, I'm afraid, he constantly says. I felt helpless, but to say, I'm here, I'll help you. It's agony watching him struggle to make sense of a world that's becoming a frightening place for him, which he can't work out where he belongs anymore. He often says, I don't know who I am anymore. He asks me, where's Jane, my wife? He asks if children present are one of ours. And then nobody talks much or writes about what happens to a sexual relationship when one partner has dementia, about the loss of reciprocity and intimacy, which can feel intensely lonely, about the transition from wife, friend, lover to carer. Spousal carers talk about living with a well-known stranger, or they often say things like, I found myself hating the person I loved, or losing a husband by slow, excruciating degrees. So still on my second point, what about this concept of the self? Now, I really struggled to understand who my husband now was. I often read that the person is still there, still the same at their core, but this didn't ring true to me. To me, my husband didn't seem to be anything like the person he was, the person that I knew. In saying, I don't know who I am anymore, he seemed unknown even to himself. The late Wendy Mitchell, a well-known person who lived with dementia, entitled her autobiography, Somebody I Used to Know, also expressing this idea that she felt detached from her old self. I came across this article written by Maureen, thank you very much, which would have been really helpful to read and understand when I was caring for my husband. In the article, she talks about the self being in flux rather than irrevocably lost. And by understanding this, we can better meet the needs of the person with dementia and the importance of us adapting to the changing personal landscape of dementia. Which brings me to something I really wanted to add to this talk. And I felt that I really needed someone who could confirm that the world as I perceived it was real to someone other than myself. And I was fortunate to come across Gaynor MacDonald, who sadly died before her work was done. Now, Gaynor was an anthropologist here at Sydney University who initiated the Dementia Reframed project. And the project aimed to reframe dementia and also reframe care not as a role, but as a relationship. Her husband had Alzheimer's disease, so she was able to draw on both her personal experience and her academic expertise in anthropology using autoethnographic methodology. She was an academic who got it. Now, I didn't meet Gaynor till after my husband died, but she really helped me to make better sense of the experience of spousal carers. She gave insights into understanding how to deal with the distress that's often experienced by people with dementia and how carers best respond to this distress. She really helped me to understand that caring is fundamentally relational. The impact of dementia on spousal relationships and the way in which the self of the carer has to change as the person with dementia struggles with their self. This slide illustrates some of the work that we did together and emphasises the unique experience of intimate partner carers and how important it is to understand this in order to appropriately support them. So now to my third point, and this is about exploring the idea of public involvement in research as grief work. So in my meanderings, which I do a lot, particularly in the public involvement literature, I came across this article, which made some sense to me and made me think a bit more about why we get involved in research, and also how important it is that the researchers understand the experiences of people who they partner with in their work. And I would argue, in particular, people who are or have been spousal carers. It made the point that patient engagement, and I'm using the author's Canadian terminology here, it's often described as the process of involving patients in healthcare research, decision-making and co-design. But there's a deeper layer rarely discussed, and that is that patient engagement is actually grief work. For those involved, it's an act of processing loss. Every time a patient shows up, they bring a layer of grief with them. Patients don't leave it behind when they participate in or get involved in research. It's with them every day. Understanding this is key to creating an environment that's truly supportive and inclusive. Further, the article encourages researchers to be open and honest about their own experiences, so as to create a foundation of trust and authenticity where patients can feel safe bringing their grief and their whole selves to the table. Showing patients that vulnerability is a strength, then in this space, everyone's humanity matters. It isn't just a show of empathy, it's a powerful way to create the inclusive, human-centred environments that truly make patient engagement work. And the author calls for a shifting of the culture of patient engagement. What if we redefine what professionalism means in patient engagement? Instead of prioritising the appearance of polish, let's focus on creating environments that are open, empathetic and rooted in authenticity. Let's build spaces that encourage people to bring their full selves, grief included. Value lived experience just as much as formal qualifications. Accept emotions as part of the conversation and not something to be suppressed. And recognise that grief is a natural part of the patient journey and deserves to be acknowledged. Again, it's not just about compassion, it's about creating effective, meaningful patient engagement. When patients feel understood, supported and safe to be themselves, they're more likely to engage deeply and meaningfully, and that's when the real impactful work happens. So I hope I've given you a bit more insight into the spousal caring experience and how this might impact on public involvement work. So just to summarise, my 3 dot points were for you to think about, and these are the chronic and often severe stress, isolation, loneliness and grief and the other emotional sequelae associated with dementia caring may exert substantial risk for the development of dementia in spouse carers and possibly death. We need to care more about carers, particularly spousal carers, and how we deal with the challenging symptoms that people with dementia may experience and the unique grief from caring for someone living and dying with dementia. And grief work is important. It is important to understand this for both researchers and those of us who get involved in research. Now, of course, we all do it for altruistic reasons because we want to contribute to the research effort and thereby hopefully improve the lives of others. But personal motivations and benefits can't be overlooked. So thank you for the opportunity to speak today, and I'll just leave you with some last thoughts, and I'm happy to take any questions.
So much, Jane. It was such a profound kind of insight into what it means to be, you know, caring for someone with dementia and also the motivations behind getting involved as well. As we call up our next speaker, we have time for one question. Someone have one. Maybe straight in there.
Thanks. It's not so much a question, I guess, Anne, but
sorry, Peggy. Are you right? You okay?
A comment. If I can, that was fantastic. Sorry, everybody. I'm a spousal carer, I was a spousal carer. Everything that you said was just superb.
Thank you.
And I really hope that the people here, researchers and so on, take very, very much to heart your words. Oh dear. Because they're just.
Do you need a hug or you?
No, that'll be worse. They're just so incredibly important. So that's all. So for everybody to do that. Superb, superb presentation. Thank you very much.
Benji. So our next speaker is Dr. Pradeep Chola, who's a postdoctoral fellow at Macquarie University, researching the role of the gut microbiome in neurodegeneration. He is currently a recipient of a very prestigious Race Against Dementia, Dementia Australia Research Foundation Fellowship, and he's going to tell us about his current work focusing on establishing a germ-free zebrafish model to study how the gut microbiome from Alzheimer's disease patients influences disease development.
Thank you. Good afternoon, everyone. Thanks to the organizers for inviting me. So let's talk about gut. So let's start with the basics and then move on. So what is a gut microbiome? The gut consists of millions of microorganisms, and together they form the gut microbiome. And we all would have faced some problems when there is an imbalance in our gut microbiome, which will include like bloating, food intolerances, or even an upset stomach. Now, just like how we breathe in oxygen and release out carbon dioxide, the bacteria in your gut does the same, and they're called as metabolites. Now, these microorganisms, along with their metabolites, have a very powerful immunomodulatory effect on various body organs. And of course, today we are going to be focusing on the relationship between the gut and the brain. It is a bidirectional communication system called as the gut-brain axis. The gut-brain axis plays a crucial role in immunity, helping us maintain the blood-brain barrier, plays a very important role in gut function, and plays a crucial role in emotional and cognitive development. Now, sometimes we have gut dysbiosis, and this might be due to factors such as diet, lifestyle factors, or even genetics. And in those cases, this gut-brain axis is disrupted. When the gut is disrupted, it creates a change in your gut microbiome, which can lead to increase in something called as the bad metabolites, which can enter your blood system and across the blood-brain barrier and have an impact on your immune cells in your brain, which is specifically your microglial cells, and cause neural inflammation. Now, this gut-brain axis disruption is seen in various neurodegenerative diseases, including Alzheimer's, Parkinson's, multiple sclerosis, and MND, or motor neuron diseases. Now, in particular to Alzheimer's disease, there's been a lot of clinical observational research and some interventional research, which has shown that the Alzheimer's disease microbiome has been involved in neuroinflammation. They actually help in aiding formation of new amyloid plaque formations in the brain, and also they are having a major impact on oxidative stress. And most of these symptoms are associated with imbalances in either the metabolites in your gut or the bacteria in your gut. Now, to study the role of gut microbiome in neurodegenerative disease as a whole, we require a good animal model to actually study it. And what do we have as of now? So as mentioned previously, yes, we can do a lot of clinical observation research, but to perform any kind of interventional research, researchers usually rely on rodent models. And rodents are mammals, which means they come with their own microbiome from the time of birth. But through stringent sterile techniques, we can convert a conventional mouse into a germ-free mouse. But these mouse come with a slew of disadvantages, including that they're very expensive, they're hard to maintain, they have a lot of developmental defects, have premature death. And most importantly, these mouse models are not malleable. That is, you will face a lot of experimental challenges when you're working with these mouse models. So this is where my research program comes into the picture, where I want to show you guys how we can use zebrafish as a model to actually study the gut microbiome in neurodegenerative diseases. Now, before I go on to explain the importance of zebrafish, I would like to assure you guys that I have used the zebrafish, germ-free zebrafish model previously to study the role of gut microbiome in cardiovascular disease complications such as hyperlipidemia. And I've also looked at the effect of smoking on gut microbiome using my zebrafish model. Now, this concept of germ-free zebrafish was developed by John Rawls, and he has actually shown a lot of similarity between the zebrafish microbiome and the mouse microbiome. And recent researchers have also shown that germ-free zebrafish is capable of harboring a lot of the human microbiome when given to them. Now, why use zebrafish? Zebrafish is a rapidly developing model. That is, by 5 days from the time of birth, they have a very functional gut, which means you can study the gut by 5 days. A single mating of a zebrafish can lay around like hundreds of eggs, which means you can have a high throughput system with which you can screen a lot of these metabolites, which we talked about. And we have developed a system with where we can develop germ-free zebrafish with ease. It's almost like having a cell culture model. And with these germ-free zebrafish, you can give whatever metabolites or the bacteria, and then you can study the effect of the gut microbiome on it. And more importantly, zebrafish are transparent. At Macquarie University, we have a range of transgenic zebrafish, which are marked for various neuronal and immune cells. As you can see in the left, you can see the red microglial cells in the brain, surrounded by the green neuronal cells. We have the capability to even zoom into one specific motor neuron over here in a live zebrafish, and you can see the branching of a motor neuron and its green nucleus. Now, because these fish are transparent, and we are imaging them live, we can almost have a live stream into what's happening in our fish. Here you can see in the brain, you can see how the green microglial cells are migrating to the site of injury. Or over here, you can see how these red microglial cells are performing immunosurveillance in your pink neuronal cells along the spine. So hopefully I've convinced you guys that zebrafish offers a lot of advantages when we are trying to study the gut microbiome. So coming back to the project, what we aim is to create a germ-free zebrafish model, perform fecal microbiome transplant. Why fecal microbiome? That is almost reflective of what happens in your gut, and then perform, which will help us create a notobiotic zebrafish or a zebrafish with a known microbiome, and with which we can actually study the immunomodulatory effects within the zebrafish. Now, to set up this research program, we started asking a lot of researchers whether we can collect some poop from their mouse. And we were able to collect fecal samples from various neurodegenerative disease mouse models, including ALS, Alzheimer's disease, and Huntington disease. And today I want to focus a bit on ALS because we have a much more completed story, and I can show you the advantages of the fecal microbiome transplant in zebrafish. Now, for one of the ALS studies, we used Delta NLS mouse, which is a very well-established ALS mouse model. And we performed fecal microbiome transplant into our germ-free zebrafish around 3 days. And within 5 to 6 days, we started observing what happens to these fish in terms of immune interactions. Now, first up, we noticed that fish which was inoculated with the female ALS microbiome of the mouse, we noticed there was increased neutrophils, which you can see in the gut, in the red dots, and also increased macrophages, but only in the fish which was inoculated with a female ALS microbiome. And there was no changes within the male cohort. Now, as you all know, that the microglial cells are one of the important cells which acts as the communicator between the gut-brain axis. So I was interested to see what kind of functionality does the microglia have in response to the fecal microbiome transplant. And I performed a targeted injury to the brain of the zebrafish. And as you would have seen this picture again, we saw how much of microglial cells are migrating to the site of injury. And similarly, we noticed that only fish which was inoculated with the female ALS microbiome had reduced microglial migration to the site of injury, and there was no changes within the male cohort. And this is suggestive that these microglial cells, or your immune cells in the brain, are dysfunctional in response to the fecal transplant. Now, another feature of zebrafish, which I want to highlight, is that we can actually drill down to the mechanism of a lot of these effects which we are seeing with the help of CRISPR-Cas9 technology, which is very well established in zebrafish. And I was very much interested in this protein called as MID88, which is an adaptive protein present in the gut. What this does is basically interacts with your microbiome and in your gut and has immunomodulatory effects, which goes to the brain and has a lot of immunomodulatory effects in the brain. So I used CRISPR-Cas9 and I knocked on this MID88 protein, and I think I got almost like 60 to 70% reduction in this MID88 expression. And I again performed the fecal transplant and the brain injury. And as you can see over here in the control CRISPR, again, you saw a reduction in microglial migration in response to the female ALS microbiome. But when the MID88 CRISPR fish had their brain injury, there was no significant reduction in the microglial migration. And this is suggesting that there was the MID88 is involved in microglial migration. More importantly, the fecal transplantation had actually influenced the microglial migration in the brain. Finally, one of the other feature which I was interested in is that at Macquarie, we have also established where we can actually observe the behavior of the fish in response to the fecal transplantation. For this, I used another ALS mouse model, which is called SOD1, which is a very well-established model as well. And for this, I actually collected fecal samples at early, mid-stage, and end stage of the disease. And as usual, we transplanted them into a germ-free zebrafish. And we use ZebraBox, which basically captures the movement of your zebrafish. And I performed something called a stigmotaxis assay or stress response. And this basically, I saw how long does a fish move in the outer ring of the circle. Now, this stigmotaxis assay is very well conserved with fish to mouse to even humans. And we noticed that fish which was inoculated with the SOD or the ALS microbiome had increased level of stress compared to its control. And this increased stress levels was only seen when we inoculated the mid-stage or the end-stage disease microbiome, and no changes were seen within the early microbiome. I had actually used this model to also see for gut inflammation, which also showed the same, a similar kind of pattern with the mid-stage and end-stage, and also for microglial function, which also showed, again, mid-stage and end-stage had severe microglial dysfunction compared to the early stage. Now, to summarize, we have performed this not just with ALS mouse models, but also with Alzheimer's disease and Huntington disease. And we found it to be consistent that there was increased gut inflammation, microglial dysfunction, a lot of behavioral abnormalities, and more importantly, we were able to distinguish the microbiome between genders, male and female, and we were also able to track the microbiome over the time course of the disease. Now, with all of this established, we have now moved on to replacing the mouse with humans. At Macquarie University, we have the Neurodegenerative Disease Biobank, with which we are collecting fecal samples from Alzheimer's disease patients, and they can range from mild cognitive impairment to amyloid beta positive patients. And we are in the process of performing fecal transplantation analysis and metabolomics on them. So what is the significance? So we are planning, at the end of this project, we are hopeful to find novel gut-directed therapies in the form of like prebiotics or probiotics that can either slow down the disease progression, and also we aim to find out unique bacterial fingerprints for Alzheimer's disease that can aid in early, non-invasive, and economical way to diagnose Alzheimer's disease. Finally, I would like to thank Dementia Australia Research Foundation and Race Against Dementia for supporting my fellowship, my group at Macquarie University, the Mosh Group, headed by Associate Professor Mark Mosh, and the rest of the team. Thank you.
We'll all agree that was a tour de force, and thank you so much. And I love your icon. Your slides are lovely. Does anyone have a question for Joe, and then
Pradeep, thank you so much for that talk. I know that there's a lot of researchers scratching their head around the world around lecanemab, and when you look at the data, it looks like it's not it's not effective for females, and they're wondering, well, why is that the case? Is potentially with the results that you're presenting here, is there some mechanism in terms of the gut microbiome, especially as you presented with the female ALS mice? I wonder, you talked about Alzheimer's disease. Is it the same for the female AD microbiome, that there is that relationship?
Yeah, So with the female microbiome which I presented, that was with the ALS mouse models. And particularly for that, we were very surprised to see that gender difference because the mouse itself showed no gender difference. So then we performed 16 of sequencing on the mouse microbiome, and the mouse microbiome clearly showed a big difference. So because that's the only sample we are getting, we're able to see that maybe because the mouse model which I showed initially was a very fast progressing mouse model. And that's why the gender differences were not seen in the mouse, but seen in the fecal microbiome. But asking your question about the female microbiome in Alzheimer's disease, I'm pretty sure it should have a major impact, especially with the hormonal changes and everything. But we are yet to, we are in the process of collecting fecal samples from the patients. So hopefully I'll be able to.
Stay tuned. Yeah, looking forward. Thanks.
Sorry, that's all the time we have for it, but thank you for deep and. All right, so the final speaker in this session is Dr. Anastasia Soreyev. All right. She's a senior research fellow at the Brain and Mind Center, where she investigates how factors associated with cognitive impairments, such as sleep disturbances and chronic pain, affect brain health and dementia risk. Her PhD focused on novel treatments for insomnia, and her current research explores whether interventions targeting sleep disturbances can support cognition and long-term brain health.
Thank you so much for having me here today. I'm delighted to be invited to speak at the symposium. So sleep is the third pillar of health, along with exercise and nutrition. And although all interact and influence each other, many experts believe that sleep is the most critical that underpins the foundation of the other two. Now, sleep disturbances are more common than people realise. Nearly half of all Australian adults report at least 2 sleep-related problems, and this rises with age to up to 40 to 70%. And the most common sleep disorders are insomnia disorder, which is difficulties with falling asleep and staying asleep, and it's associated with daytime impairments. And it affects about 1 in 10 people in the general population, and it's usually predisposed towards women. The second most common sleep disorder is obstructive sleep apnea, which is a sleep-related breathing disorder, where the airways are obstructed during the night, resulting in less oxygen getting into the blood and into your brain. And men tend to be more predisposed to sleep apnea. Now, sleep disturbance negatively impacts our daytime function, how well we're alert, how well we can concentrate, our cognitive performance, and also how we feel, so our mood. And over time, it can also contribute to chronic disease. So we know that sleep disorders and sleep disturbance can increase our risk of depression, cardiovascular disease, and also diabetes. Now, sleep disorders are common in aging. As I mentioned, the prevalence increases as we age, but it's also even more common in neurodegeneration. So in this table here, we highlight 3 different sleep disorders. So insomnia, sleep disordered breathing, and REM sleep behavior disorder. And for example, while insomnia affects around 9 to 10% in the general population, this increases when you're looking at all-cause dementia to 27 to 52%. On the other hand, REM sleep behavior disorder is not very frequent in the general population at 1%, but is quite prevalent in certain types of neurodegenerative diseases, including Parkinson's disease and dementia with Lewy bodies. So it's very strongly linked in these conditions. And interestingly, more than 1/3 of people with mild cognitive impairment, which is the high-risk stage for Alzheimer's disease, report sleep disturbance. So there is a clear relationship between sleep disturbance and neurodegenerative disease. So what does the research show about the link with dementia? So this is a meta-analysis of 18 longitudinal studies with almost 250,000 individuals at baseline, and they followed them up for almost 10 years. And of those, a number went on to develop dementia. And they showed that individuals with sleep disturbance, self-reported sleep disturbance, had a higher risk of incident all-cause dementia and vascular dementia, and also Alzheimer's disease. And so what this suggests that even we can't say that sleep disturbances necessarily directly cause dementia. What we can say is that it can help us identify individuals who may be at risk for dementia down the track. Now, it's not just about the quality of your sleep, but it's also about how much you sleep. So this study looking at individuals over 25 years, so almost 8,000 older adults, of which 521 developed dementia. They showed that the lowest dementia incidence was observed among those who slept for 7 hours a night. So as you can see on the graph on the right, you've got that sweet spot in the middle and presenting with a U-shaped relationship where short sleeps, so less than 6 hours of sleep, or long sleeps, anything over 9 hours of sleep, was associated with a greater risk of dementia down the track. And this remained significant after adjusting for socio-demographic factors, health behaviors, cardiometabolic and mental health factors, and a subset of this was also confirmed using actigraphy. Now, sleep duration is an interesting one, and so that was brought up earlier this morning in terms of, is it a risk factor, is it a prodrome, so is it a consequence of the neurodegenerative process? And this study recently published showed, because it's a meta-analysis looking at all the studies that have looked at short sleep or long sleep duration, and what they found was that short sleep was linked to higher dementia risk, but only in studies with shorter follow-up periods, less than 10 years. So it may suggest that short sleep is a prodrome symptom rather than a direct risk factor. Long sleep, on the other hand, was consistently associated with increased dementia risk, and that was both short and long-term follow-up, and may be a direct risk factor. However, they do note across all the different studies that were included in the meta-analysis that there was quite a lot of heterogeneity in the results and also a high risk of bias. So we can't yet sort of generalize across all our other areas. So we do need more research to determine using objective sleep measures, and also longer follow-up periods are needed. So what we can conclude is that sleep disturbance may be a consequence of neurodegenerative disease and also contributed to. And certainly the research in the field currently is trying to explore that relationship. And we certainly need a wealth of information, consensus, as well as the biobank data to really hone in on what that relationship is showing. So before I move into the next slide, the way that we can understand whether sleep is an important risk factor, or to understand if it's something that is can potentially, if we treat it, can improve cognition down the track, is to run clinical trials. So what I want to do is give you an overview of all the current clinical trials, both pharmacological and non-pharmacological, that are looking at sleep interventions for people, older adults, and also people with mild cognitive impairment. So starting off with non-pharmacological sleep interventions. So cognitive behavior therapy for insomnia is the gold standard for treating insomnia disorder. And it's certainly shown to be effective in adults with insomnia disorder. And we do have preliminary evidence for both groups, so having a group setting and administering the therapy, and also digital, so an online therapy of this CBTI in improving sleep, but also subjective cognition in people with MCI. What we haven't yet shown is whether there are long-term cognitive benefits with CBTI, and so we certainly need larger studies in this population to show those changes. Exercise is an important one, so thinking back to the 3 pillars of health, certainly there is strong and good quality evidence showing that exercise can improve both self-reported, so how you feel, sleep, and also objective sleep, both in people with mild cognitive impairment and Alzheimer's disease. What we haven't yet done is that next step in showing how does it then affect cognition? Does it improve cognition, or does it halt cognitive decline? And how does it affect brain metabolism? So there are several ongoing trials that have been pre-registered that are now shifting focus and looking at that longer term trajectory. Bright light therapy certainly has been explored extensively, particularly in patients with Alzheimer's disease and also in care facilities, but not very much in people with mild cognitive impairment. So looking at that sort of early intervention phase, and certainly the existing evidence is equivocal at best. And so certainly more studies are needed there. There is some preliminary evidence showing that morning light exposure may result in less fragmented sleep. So in training your circadian rhythm can help improve your sleep. CPAP therapy, I think it was also touched on earlier this morning. So there's been large meta-analyses showing modest improvement in sleepiness and attention in people with sleep apnea. And a systematic review looking at over 60,000 individuals with sleep apnea showing that CPAP therapy, which is the gold standard treatment for sleep apnea, had a protective effect on mild cognitive impairment and Alzheimer's disease incidence. So certainly, a really important therapy, and really focusing now on how to ensure adherence to treatment, as that is a particular concern, particularly in people with MCI and Alzheimer's disease. Pharmacological intervention. So the first thing I want to say is that benzodiazepine and Z drugs are generally not recommended in older adults. And this is mainly due to their adverse effects and so things like heightened risk of falls and injury, increased risk of drowsiness the next day. Melatonin is a safer alternative, and it has modest efficacy for insomnia and also circadian rhythm disorders in older adults. However, we don't have good long-term safety data. So anything greater than 12 weeks, a lot of these studies have focused on 3 months and no longer, except in the exception of this one study where in patients with Alzheimer's, they had ongoing treatment with a low dose of melatonin, and that showed better cognitive performance after 6 months. That's very interesting and promising, but we still need more robust randomized controlled trials to show benefit. And this is not new. It's certainly not an emerging drug. It's very much been explored for treating insomnia disorder. What a lot of these companies have done, so orexin receptor antagonists promote sleep by blocking the orexin receptor. And so the shift has been looking at how it affects older adults. And so there's certainly mounting evidence showing that it is safe and effective in older adults. There's benefits to sleep onset and maintenance. There's less nocturnal awakenings. There is some risk with daytime sleepiness, and particularly at higher doses, so very much kind of something to be mindful of with these drugs. But a really interesting shift here is looking at potential neuroprotective effects of these drugs. And these are being explored in ongoing trials and looking at the effects on cognition and also Alzheimer's disease biomarkers. So we just summarized a lot of that literature recently in this publication, looking at both current and emerging sleep interventions for older adults, and trying to understand where are we going next. And really the biggest shift that we've noticed in the literature is looking at how these sleep interventions can improve or halt cognitive decline, and also looking at how they affect brain metabolism, Alzheimer's disease biomarkers, really trying to predict can changing and improving your sleep have an impact on your brain health. Now, for the last few minutes of my talk, I just want to talk about 2 new studies that we're working on in the Healthy Brain Aging Clinic that are looking at 2 key sleep disruptors. So for those of you that you don't know about the Healthy Brain Aging Clinic, we are a specialist memory clinic at the Brain and Mind Centre, and we invite individuals from the community who have new onset concerns about their memory. And so individuals will get a GP referral to come and see us. They will sign in consent and complete a series of self-report questionnaires. They will then come to their appointment where we'll collect some blood. From there, we can do APOE genotyping. We can assess for neurodegenerative biomarkers, neuroinflammatory markers. They'll then complete a medical assessment with our geriatrician, a full neuropsychological assessment, and also a mood and sleep assessment. And all of this together will help inform diagnosis between the clinicians. After the appointment, we'll also offer a range of other assessments. So we'll get a clinical bloods referral. They'll complete an overnight sleep study. They'll take home some wearable devices to monitor their sleep and also how much oxygen is getting into their blood overnight. Some will also go on to do brain MRI and PET. And so PET will look at the amount of amyloid burden in their brain and arterial stiffness. And then also, if they're eligible, some people will also be referred on clinical trials. We also recommend that they come back in 2 years' time. We want to see how they're doing. And so up to 4th visit, they'll come back and we'll do the assessment again. So the first study that I want to touch on is a comorbid condition that affects so many people in Australia and worldwide. 1 in 5 Australians experience chronic pain, and also 75% of them report sleep disturbances. And we know that pain and sleep both exacerbate each other. So if you don't sleep very well the next morning you'll have increased pain sensitivity. And then if you don't sleep very well, sorry, if you have really bad pain, then it's more likely that it's going to cause some sleep disruption. And they both place a very excessive burden on the brain and cognition, and are linked to higher disability, depression, and also increased healthcare service use. And so what we wanted to look at is the relationship between both chronic pain and sleep disturbance. We know that's a very, has a strong bidirectional relationship, and that's well established. But we want to know, how does this relate then to cognitive impairment? And there's been multiple studies now showing that people with chronic pain have accelerated brain aging. There's an increased evidence of brain atrophy and also cognitive impairment on testing. So we want to look at that relationship in greater detail. So our PhD student, Anastasia Saramovska, will look at how we can treat and will explore this relationship further in our cohort. So the first study will look at the relationships between these three things in our cohort. She's already explored the existing data set and looked at those individuals who had pain versus no pain, and found that poor sleep significantly predicted pain in older adults with mild cognitive impairment, but not in those with subjective cognitive decline. And so this suggests that poor sleep is a key pain predictor in mild cognitive impairment. Her next study will then go on to determine which components of digital cognitive behavior therapy for insomnia, which is the gold standard treatment for insomnia, are effective in improving pain in individuals with comorbid chronic pain and insomnia. So she's doing a systematic review and component network meta-analysis to determine and inform the co-design of an adapted manual for chronic pain. And finally, this will be a co-design which will engage with stakeholders, pain specialists, people with lived experience and living experience with dementia, to test this and before we go into a feasibility RCT and determine what this data shows and how we can move it forward to a definitive clinical trial. Now, I realize I'm going a little bit over time, so I might just skip up the second study. I do just want to quickly note that the second study looks at menopause in women. Women are twice as likely to develop Alzheimer's disease than men, and we actually still don't know why. This was a Nature paper that was published just this week. And so one of the important things that we're doing in the clinic is trying to explore this relationship. And we believe that it may be due to the changes that happen during menopause in women. So I will very, very briefly touch on this project that our PhD candidate is working on. So looking at sex differences and menopause transition in older adults and how this relates to cognition. So very briefly, just going to be looking at a range of different things, looking at sex differences in our cohort, how menopause transition is associated with cognitive impairment, and then how these hormone levels then are associated with changes in kind of the trajectory in terms of cognitive trajectory. The final key takeaways, sleep disturbance is a key feature of neurodegenerative disease. It serves as both a risk factor and a consequence of the condition, but we do need a lot more data to support this and better understand this relationship. And finally, there's lots of emerging priorities that are coming out of this research, and chronic pain and also menopause are very key, I guess, research areas that we're interested in. So thank you so much for listening. And you're welcome.
Fantastic. I think we'll leave questions until the end for Anastasia, but please tag all the speakers in this session.
Moving into the next session, I'd like to welcome the first of our 3 rapid fire round speakers, Dr. Jacqueline Weston. So Jackie is a senior occupational therapist and early career researcher. Functional cognition is foundational in her work, examining the impact of cognitive impairment on performance of everyday activities in older adults across the continuum of ability, from very subtle changes to more severe impairment. Welcome, Jackie.
While Shannon's getting the slides up, oh no, she's already done it. Excellent. Thank you for the opportunity to talk about the Research Impact Award that I got as an ECR. I'd like to give you a quick overview of one of the programs of my research and just to illustrate how it's having an impact, and it's very much a a bird's eye view. So I'll talk to you about functional cognition in older adults, detecting change and supporting abilities, which I think these ladies are doing both of. So as an occupational therapist, I'm thinking about what the clinical conundrum is here. What is the impact of cognitive change on functional performance? So in my role in the memory clinic, I saw people that would have no impairment on neuropsych testing, but talked to me about functional difficulties. And these were usually during complex everyday activities, often leaving them quite addled about how things ended up the way that they did. And so this got me thinking about my role as an OT. I always am looking and assessing on how people explore their everyday, or how they perform their everyday activities, as shown in the grey box. So I'll talk you through this somewhat complex looking slide, but it's, so I'm at the top, I'm looking at function and then inferring on what might be an impairment or a deficit, and this being referred to as a top-down approach because we're using the occupation as the assessment medium. And then it's a performance-based concept, so it's looking at how people are using cognitive abilities in the moment while they're doing their activities. And so that's quite different from a traditional cognitive approach where we can assess a cognitive domain quite discretely, usually with pen and paper tasks or something that's a little bit more proxy simulation of real life activities, but it still has that more discrete approach to the cognitive domains. And that would be referred to as a bottom-up approach where we're looking at discrete areas of difficulty and then inferring up to think about how that impacts on function. And so we know that both of those approaches are really important. It's not that one's better than the other. They actually complement each other and we need them both. But what I really needed was some evidence that this functional cognitive viewpoint and this perspective that I had was actually a thing in clinical practice. So this is where my PhD took me. And these papers are the first two of my my PhD, and where the impact has really come from. And surprisingly, it was an unpredictably positive impact, probably because I didn't have a research impact plan at that point. I didn't know that was a thing. So what I did was defined functional cognition in that first paper in the systematic review. And you can see there are a number of unsolicited invitations that came out of that to write a couple of book chapters. I, like Jane, also wrote so much that they turned it into two. And also a couple of PhD candidates. And I now run an undergrad course for OTs here at Sydney Uni. And at the moment, there's a new initiative just recently invited to join an international group of OT professors looking at a conceptual analysis in the literature of functional cognition. I also applied innovative methodology in that review. It was a review of assessment tools. And then that's now led to my expertise in methodology being sought through a range of other projects, some of which are mentioned there. And of course, the clinical impact. And this is emerging. It's still a work in progress. So I'll just talk you through that. So that second paper that I mentioned, the validity study, really was the first evidence for OTs in Australia that a functional cognitive tool was able to discriminate between people with healthy cognition and people with dementia. And that combined with my other sort of areas and projects that I've been working on with other collaborators as an AI or a CI on projects, led to the DARF project grant, which is currently underway. And so, of course, I'm obviously looking for grants, so I need to hand any of those out to you. But what we're doing there is looking at the lacing task, which is that validity study that I talked about and where my sort of origins of functional cognition came from. It's an OT model. and also comparing that to the menu task, which is another screening tool. And I've introduced that to Australia. It's been validated in the US. So it's the first time, you know, fun, exciting things like changing the menu and making sure it's an Australian menu that we are offering our participants. And I would just like to finish off by saying that that's a work in progress very much. I would like to acknowledge all of my supports along the way, that there's no way that I could have potentially been where I'm at the moment with this project up and running without people like Henry Bradati and Simone Repperman, who were supervisors for my PhD at the time and are currently on that DARF project grant. It's leveraging a bigger project that Simone's running on computerised IADL. But if I would also like to acknowledge Peggy, who's one of my research advisory group members and features strongly in the video. So if you want to have a look at that, please do. And really that's all I have to say. So thank you very much.
Thank you, Jackie, and congratulations again on your award. Well deserved. Next up, we've got Dr. Samuel Warren. So Dr. Warren is a computational neuroscientist at the University of Sydney. His research focuses on ways to diagnose dementia using neuroimaging, neuropsychological tests, and machine/deep learning. Welcome, Sam.
Oh, easy. Sorry, I hope you don't mind. All right. So hi, I'm Sam, as told. I'm a postdoc here at BMC and University of Sydney, specifically part of the frontotemporal dementia research group and parish lab. And today I'm just talking about neuropsychiatric conditions in frontotemporal dementia and ways we can kind of distinguish types of frontotemporal dementia based on these neuropsychiatric symptoms. So first off, neuropsychiatric symptoms are prominent in all types of frontotemporal dementia. And these kind of symptoms are highly heterogeneous, meaning that they're highly variable in each type of frontotemporal dementia and then across the different types of frontotemporal dementia. And this heterogeneity can be quite problematic because it can result in disease misdiagnosis, so within FTD types, but also with things like Alzheimer's disease and psychiatric conditions. And it can also result in poor prognosis, meaning that it's hard to understand where someone is in their disease and where they will be. So this is a study done by Altamir et al. Essentially, they looked at neuropsychiatric conditions, symptoms in 3 different types of FTD. So behavioral variant FTD, semantic dementia, and progressive non-fluent aphasia. And essentially, they just plotted out the frequency of these neuropsychiatric conditions in these FTD subtypes. And as you can kind of just visually tell, across this kind of gamut of NPS, there is a high frequency of them happening in each of these conditions. And even in these conditions that we would usually define by a specific NPS, for example, like apathy at the end, it's still quite common in all 3 of these types of FTD. So what my job kind of is to look at this heterogeneity and look for ways that we can kind of parse it and kind of make meaningful groups and differentiate diagnoses. And the way I'm doing that is through clustering. So clustering is essentially just a machine learning technique. And what it does is it groups people based on relationships in their data. Okay. Importantly, it's transdiagnostic. So it's looking at everyone in FTD. It's not looking at specific types of diagnoses. So I specifically used latent class clustering analysis. And then I used a retrospective sample from NIFD, which is a very big FTD database in the US. And the sample had 67 individuals with BVFTD, 39 with SD, 32 with PNFA, and then 81 controls for comparison. And these were all at baseline. So the idea is to kind of look at how these people are presenting in the clinic and then see if we can differentiate them based on these neuropsychiatric symptoms. The specific variable I used was the MPI, which is a very famous neuropsychiatric kind of screening tool for dementia. And then I also controlled for age, education, and cognition, just meaning I held these kind of constants so that they weren't influencing the results. So this is what the results look like. Essentially, the model pulled out 5 different groups or 5 different clusters of kind of these psychiatric profiles. And then on the left going down, you have the actual diagnosis defined by the clinic. So you can see in cluster 1, it is predominantly semantic dementia and behavioral variant frontotemporal dementia. And the next cluster is kind of this mixed FTD group. The next is very clearly kind of these controls. The cluster 4 is predominantly BVFTD. And then the last one is this very interesting PNFA and control group. So then what we can do with the model here is we can plot out the actual profiles of neuropsychiatric symptoms in these clusters and use that to look at these kind of group differences. So this is what it looks like. Hopefully it's not too scary, but essentially each colored line is one of those clusters, one of those groups. On the left, you have the probability of them having a psychiatric condition, and then along the bottom, it has the actual kind of psychiatric conditions in the, symptoms in the MPI. So starting from the top, we have that predominant BVFTD group, and you can see that they have a very high probability, more so than any other cluster, to have neuropsychiatric symptoms. And they have specific peaks in things like appetite, disinhibition, irritability, and like motor, like those repetitive behaviors that we expect in BVFTD. Next, we have the yellow group, which is that semantic variant and that behavioral variant mixed cluster. And you can see that they follow a very similar probability, a very similar MPS profile for these symptoms. It just, the probability is a little bit lower. Next is that mixed FTD group. And here you can see that once again, very similar profile, but the probability of having these psychiatric conditions is a lot lower. And then there are some deviations though that start to occur. For example, in motor at the end, it doesn't seem to be as big a thing as in those more BV kind of clusters. And then last of the symptomatic ones is the PNFA group that was mixed with controls. And you can see that kind of as expected, being mixed with controls, they have this very low probability of neuropsychiatric conditions. But there is still that symptomatology and that kind of very similar profile of what you're likely to see in those groups. And then you got controls. So what does it all mean? Essentially, clustering can be a way to kind of transdiagnostically pull out these MPS profiles in different groups and kind of meaningfully use that to distinguish these conditions. So although there is still heterogeneity, it is still easy to kind of see that there are diagnostic similarities in these specific clusters. So we observe this kind of clear relationship between clusters and the frequency or the occurrence of neuropsychiatric symptoms. But what we really find is that the specific MPS is not so much important as kind of the profile or the chance of having an MPS. And I think one important thing to stress is that while kind of these clusters and the way I present them is that they do have a dominant diagnoses, there is still heterogeneity. There is still someone with any type of FTD in each cluster. So I think it's really important to kind of look at MPS profiles altogether, but also look to not kind of diagnose people just on one specific neuropsychiatric symptom. And in the future, the plan is to kind of expand this approach to other conditions like Alzheimer's disease and psychiatric conditions to see how they connect and compare to FTD, and then also kind of validate this more in a local sample like the Frontiers group. Cool. And yeah, thank you so much.
Thank you, Sam. I now have the pleasure to invite Madalena Shibila, which is a postdoctoral research fellow from Macquarie University. And her research is focused in developing and testing innovative treatments for neurodegenerative diseases and using advanced models to deepen our understanding and how these conditions progress. So I'm going to leave it with you.
So hello, everybody, and thank you so much for the organizers for inviting me today to present some of our work. So I will talk about one of our projects, which is how amyloid beta precursor protein facilitates tau propagation in Alzheimer's disease mouse models. So Alzheimer's disease is a progressive neurodegenerative disease and the most common form of dementia. And we know that it is characterized by the deposition of two main proteins, which is amyloid beta, that results of aberrant cleavage of the amyloid beta precursor protein and then forms extracellular plaques, and the tau protein, which in disease gets hyperphosphorylated and then deposits in neurofibrillary tangles. So far, a lot of research has actually focused on how these two maybe act in tandem to progress towards dementia, but not a lot of research has been looking at what the role of APP could be in that context. And that was something that we wanted to investigate a little bit further. And I guess the inspiration to our project came from a study by Takanashi and colleagues who used HEK cells, which they transiently transfected with APP, and then they added fibrils to the media. Yeah, so I'm in this panel. And what it actually showed was that tau was internalized into these cells, suggesting to them that APP could actually act as a receptor for tau. So why is that important? Well, that would mean that APP could also be involved in tau pathology spreading, which we know is actually correlating with disease severity and progression in Alzheimer's disease. So in order to look into this a little bit further, we were asking whether APP was actually modulating tau pathology. And to investigate this, we employed a method that is quite commonly used, which is injecting human AD extracts, vehicles, or controls into tau 58 mice. I don't know why it's progressing so quickly. Into tau 58 mice, so either in the presence of APP or in the absence of APP. And then 2 months later, we wanted to look at how much tangles have formed in these mice. So when we looked at tau 58 mice in the presence, sorry, it's just jumping a little bit, in the presence of APP, we saw that mice that were injecting with the AD constructs had actually quite a progressive amount of tangles in their hippocampi. Whereas when you look at the controls or vehicle injected mice, this was not the case. What was even more interesting to us was that in the absence of APP, this was actually significantly reduced, which is quantified here on the right, and which suggested to us that APP seems to be required for the induction of tau pathology. So to now look into tau spreading and whether APP was playing a role here, we used an AAV approach. So this AAV is based on two different products. So it's GFP and mutant tau that are combined with each other through a linker. And so mice that are injected with this AAV, so neurons that are transduced in these mice with this AAV would present both GFP and tau, so so-called donor neurons. But once tau starts to spread and is taken up by a recipient neuron, this neuron would then be only positive for tau. So in our stainings, it would appear red. So this is quite kind of the first method that allows us to distinguish between viral spread and tau spread itself. So to now look a little bit further into the role of APP in this context, we injected this construct into 4 different animal models. So one model in which APP was present, so our APP plus mice, APP mice, mice in which APP was deleted, and then 2 of our Alzheimer's disease models. So APP23 are overexpressing mutant APP, and APP knock-in mice are a humanized model harboring 3 mutations. So we then wanted to look at how much tau has actually spread into these mice. So interestingly, what we found is that in the presence or within our AD mouse models, we had actually a significant amount of tau being spread. Whereas when you compare this to increased levels of APP, so in our APP plus mice, this was reduced, and even more so in our APP depleted mice, which is quantified here on the left. And this suggested to us that APP was actually involved in tau pathology spreading. So to summarize these findings, so we found that APP was facilitating the induction of tau pathology and promotes the spreading and potentially also the secretion of tau pathology. Our data also showed that this process is significantly increased in the presence of APP mutations and also increased levels of APP, so in our APP plus mice, for example. So in summary, our data suggests that APP is an integral part of tau pathology spreading and further delineates a novel role for pathogenic APP mutations in AD. Thank you very much. And I would like to acknowledge the whole team that has worked on this study, in particular my supervisors, Janet and Lars Ittner, all our collaborators and our funding bodies. Thank you so much. So for the final session today, we have a panel discussion on the topic of frailty and dementia. So I'd like to invite all our panelists up so we can hook you up to the microphone. Yes. Lots of room. That's it, I'm being pictured. What's that? Okay. Over up here, get it down there. So it's really hard. Yeah. Yes. So she feels so nice. We need to do anything with this when the town comes. And they'll unmute it. No, it's not something. What's that? All right, so we'll start. Hopefully everyone can hear us. So we're joined today by 4 experts in the topic of frailty and dementia, and they come from quite various backgrounds. So hopefully we'll have some very interesting discussion and have some unique perspectives on this topic. So we have Dr. Peggy Tromp, who has had a very long and varied working career. Peggy's husband recently died as a result of frontotemporal dementia, so she has had many years of experience caring for him. She's a member of the invaluable support group for frontotemporal dementia, auspiced by Dementia Australia, and she's a member of 5 advocate and consumer committees focusing on dementia research and is a Dementia Australia advocate. So she'll be giving us a lot of perspectives on the care and also a consumer perspective on frailty and dementia. We also have Professor Andrew Philp, who is the head of the Biology of Aging program and convener of the Centre for Healthy Aging at the Centenary Institute. And he's an internationally recognised expert in studying mitochondrial metabolism, with particular focus on skeletal muscle wasting disorders, such as aging, muscular dystrophy, and mitochondrial myopathy. And in terms of frailty, will be giving us some perspectives on sarcopenia and the pathophysiology behind all of that as well. We also have Professor Ruth Hubbard, who holds dual clinical and research leadership roles as Director General and Geriatric Consultant Geriatrician at the Princess Alexandra Hospital and Masonic Chair of Geriatric Medicine at the University of Queensland. She's the founder and director of the Australia Frailty Network, and her team
working together to answer critical questions relating to the measurement and management of frailty. I'm very much looking forward to hearing more about her research. And we have Dr. Bonnie Liu, who's a geriatrician at Royal North Shore Hospital and a PhD candidate at the University of Sydney. Her research looks at the clinical application of the drug burden index in optimizing pharmacotherapy for older surgical inpatients, and her clinical work as a general geriatrician as well as in perioperative medicine will give us further insights into real-life management of frailty and dementia in hospitals. So I'll pass on to Peggy first to start with a discussion and give us some insights into her role as a carer and consumer.
Thanks very much, Helen. Is that on now?
Yes.
And I'm going to stand up anyway because I'm just going to start off with a little personal story against myself in a sense, and hopefully this will be a bit more cheerful than what I was a little earlier with Anne's wonderful talk. So you have to picture this because we don't have slides, but imagine I'm sitting in a chair that is like a recliner, only it's a pretty crappy one. Okay, it's not exactly an excellent recliner. This is a few years ago. I'm recovering from 18 months of very severe chemotherapy due to a mastectomy, breast cancer. I've had a shoulder replacement. The other shoulder is affected with arthritis. Both the knees are pretty rubbish, and so I'm just lying back there. It's about midnight, and I'm thinking I better go to bed. So I tried to get out of this recliner thing. There's a button on the side. I press the button and to lower the whole thing, I'm leaning forward, but because this chair is not a very good one, the whole thing slides forward. So I'm now in almost this sort of a position. So what to do? I've no upper body strength. I can't push myself back up. The legs are no good, the knees are no good. So I think, righto, I shall slide gently down onto the floor and perhaps I might be able to get up from there. So I do that and then I discovered, no, can't possibly move again. Everything's not good. Okay, well, in that case, it's too late to ring anybody. I will, and I'm living alone. What will I do? My husband's in a nursing home. I'll just sleep on the floor. So I get a pillow, I get some blankets down, and I'm thinking I'll do that. And then in the morning, I'll ring somebody. However, I do that, and the first thing I think is, oh my God, I haven't seen a cockroach all summer. I guarantee there will be one. And as I'm saying this, the first one appears coming toward me. So I bash on it, not it, on it couldn't possibly. Bash the floor and it runs away. It's a timid cockroach. But it's run away and then the second one comes out. But it's a very bold cockroach and it keeps coming toward me no matter how much I bash on the floor. So it just misses me but goes underneath my cushion. God, what can I do? Ring the ambulance straight away. Ring the paramedics. They turn up. Long story short, they're wonderful people. They finally get me up. Make me a lovely cup of tea. They're very glad to be there. They love the dog, so everything's fine. Am I frail? Yes or no?
Yes.
I'm frail, aren't I? Right. I don't think I'm all that frail now. So in other words, frailty is not a continuum. Okay, number one. So one can be very frail and then one can recover and be not so frail, even though one is of a certain age. I can tell you that. So I guess coming back to the more serious side of things, I guess my point is that sometimes diagnostics, we need to take a fairly flexible approach to what frailty is or isn't. So that we know that frailty is a medical condition and that it's characterised by increased vulnerability to stresses and which may lead to a higher risk of poor health outcomes. So it's often associated with ageing, but it's not definitely solely defined by age. Young people may be frail, obviously, for all sorts of reasons. But there does tend to be an emphasis on age frailty. Old people are frail. seems to be the, general sort of view. But however, it is a distinct clinical state and it's involved in a decline in physiological reserves and resilience. And it'll be interesting to hear, Professor Andrew Phillip talk about his work. So lots of definitions exist, but one widely, and I will now sit down, one widely accepted description comes from the Frail Older Persons Collaborative, which defines frailty as a state of increased vulnerability. We also use a thing called the Clinical Frailty Scale, which people may or may not be familiar with. It categorises individuals into 9 different levels, ranging from 1, very fit, in the middle around about a 5 is mildly frail, where you need help with higher order daily activities, and ending in 9, which is terminally ill, where the person has a life expectancy of less than 6 months. Now, the scale does help clinicians and caregivers assess frailty and plan appropriate interventions. But the thing is, it also emphasises that frailty isn't about age alone, but it's about function and resilience. So frailty and dementia often coexist, but it's a bit chicken and egg, really. Does one become frail because one has dementia, or does frailty arise because of it? So obviously people with dementia are more likely to be frail due to cognitive decline and motor function decline and so on. And at the same time, frailty can worsen dementia by increasing susceptibility to infection, to hospitalisation and to other complications. My husband had behavioural variant frontotemporal dementia, and he became, at home, he was still had motor ability, but he was hospitalised at one stage for 10 days due to influenza A. At the end of that 10 days, I was advised that he would have to go to a nursing home because his mobility had gone. So from being able to walk 10 days later in hospital, he was no longer able to do so. The home wasn't big enough to be able to get him into the shower, for example, in a wheelchair and that sort of thing. And so very reluctantly, I had to place him in a nursing home after looking after him for 2 to 3 years at home. And 5 years later, he died. But it was just interesting to see that there had been no intervention there. Had he been able to have perhaps appropriate physiotherapy during those 10 days or whatever, then perhaps he might have been more mobile and be able to stay at home for a little longer, who knows. So I guess when we're looking at diagnosing or identifying frailty, there are obvious challenges. I've already said that frailty is rarely discussed in younger individuals, despite the fact that it can affect them. The frailty scale itself has got little diagrams on it, and apart from the first 3, where the little diagram, the little person appears to be relatively upright and so on, it very rapidly morphs into a sort of astute, clearly older individual until finally, we've got this person who's in bed and who is clearly the little symbol looks very much like quite an old person. But the other thing too is that the term itself carries a stigma. So I did some very rigorous research just recently, asking 6 of my friends how they felt about dementia, how they felt about frailty, Did they consider themselves frail? Well, they told me to frail off, so, in no uncertain terms. So it is indeed a loaded term. So, and as I said earlier, you might be classified as frail or moderately frail or reasonably frail, as I was, but only to regain strength later and be reclassified as managing well. So it is a bit of a chicken or egg situation as far as my view is. It's a state of increased vulnerability, and it's distinct from but often associated with ageing and dementia. So what comes first? Dementia or frailty, which is which? And I think it is very much a chicken and egg situation. So the frailty scale itself, while it provides a practical framework for assessment and care, the interventions have got to be tailored to account for perceptions, different perceptions, as I've already said, but certainly cultural interventions. We're not thinking about that. It's not mentioned, and also age groups. So I think that's about it, and thank you for your time.
Thank you for sharing your experience and also your experience as well. And it's something we see so often in hospital, deconditioning and this disproportionate change in function following a hospital stay. And I'll pass on to Andy to make some comments about work in sarcopenia and the elderly.
Sure. Thank you for that. That was awesome. So yeah, my basic background is a clinical exercise physiologist. So I studied muscle adaptation. So I started looking at athletes and then transitioned more into clinical scenarios. And the reason I love muscle is it's always adaptable. So even in your 90s and 100s, you can still improve muscle strength, muscle size, and muscle function. So there's no inevitability of muscle decline. We know sarcopenia is a disease of aging, but it actually starts in midlife. So from our 4th decade of life, we start losing strength and size of muscle. And you don't actually appreciate that in our 40s, but we associate that with an older, frail, end of life process. So I think what that tells you is actually we can really change how we age by just thinking about things earlier than later. But the most dramatic thing that happens or drives muscle loss is inactivity. So that scenario of being in a hospital bed is really the devastating thing that accelerates that loss. So we've done studies where we've taken volunteers, so healthy males in their 70s who volunteered to go into bed for 5 days. Imagine that, lots of volunteers. The problem is in that period of time, they lost about 2 kilos of muscle mass. So the next time you're in Woolies shopping and you're looking at your food, that's about 4 chicken breasts that you'd buy or 4 steaks. It's a huge amount of tissue, like an extortionate amount of tissue. And if we actually look at natural aging, that's about 3 years of natural aging in 5 days. And that is the difference between coming out of bed and not. So we know nothing about, well, we know something about what drives that, but we can't prevent it. There's no real drugs to stop that rapid loss. The problem is if you do come out of bed, you're subjectively weaker than when you began. And the critical thing with age, so that in itself is not an aging associated thing. So if you're a 20-year-old, you still lose that muscle. The difference is you get it back a lot quicker. So with age, we lose resilience, or we lose our ability to regain that strength and that size. It takes about 3 times as long. So a younger 20-year-old would recover that in about 4 weeks with training. An older adult is probably about 12 weeks to 15 weeks. And that's with strict training. So if you don't have that, then you come out of hospital subjectively weaker, and that leads to a fall and a fracture and something a lot more devastating. I think for us it's trying to understand that loss, but actually thinking if we prepare for that, we can also prevent it as well. So I love the chicken and egg scenario. I certainly can tell you that muscle loss happens a lot earlier, probably in dementia. I'm not saying it's causing it, but I do know we can target muscle. We can actually do something about that. So for us, we write lots of grants saying, how about this? If we promote muscle growth, do we protect from dementia? We'll never get that funded because no one will ever fund that kind of study. Or can we stop cachexia, or can we do other things? So this idea of prehabilitation, can we prepare our bodies for that insult? But to me, I think that's the question we're interested in. What is it about muscle that's protected to the brain? And if we can understand that, maybe that helps us to actually understand the dementia as well. So I will stop there.
Thanks, Andy. And so you mentioned how quickly we lose the muscle. So in a hospital scenario, what would you suggest be the minimum amount of time that people be out of bed and walking to prevent the muscle loss while they're in hospital?
Well, most geriatricians I speak to will get people out of bed as quick as possible. So, and if you can do that, fantastic. We're actually, I mean, some of the molecular signals in muscle happen within minutes. So, if someone came into the hospital, admitted, went overnight, and then saw someone the following day, probably within that 24-hour period, they've already started losing muscle. So it's so quick, it's so rapid, and we don't know how to stop that. But movement, no, muscle wants to move, it wants to adapt. As soon as you take that away, that drives that inactivity. So the quicker, the better. And that's just obviously loading the tissue. But I think elective surgery is very different because you can actually prepare for it. And there's some lovely studies looking at prehabilitation to improve muscle before you have that period of inactivity. If it's unplanned, then you can't prepare for it. So, but I think certainly being out of bed and moving is, at a minimum, is going to start activating and contracting that muscle, which ultimately is what stops it wasting.
Thank you.
We should all stand up, shouldn't
Yeah, like coming back to the chicken and egg question, Ruth has done some very interesting research on frailty and dementia, and the notion that frailty is a consequence of dementia is really being challenged by your research.
Yes, so I want to start by thanking the organisers for inviting me down to the panel today, and I'd like to acknowledge Peggy's authenticity in her presentation today. It's quite unusual for me to be on a panel where somebody else has said what frailty is, and spoken about all the different ways it can be measured and the kind of core definitions of frailty. But I would like to challenge the group today by proposing that frailty isn't just a consequence of dementia, but maybe something that can increase your risk of developing dementia and manifesting the symptoms of dementia in relation to neuropathology. So the first case of dementia was described by Alois Alzheimer's. I'm sure you all know. She's probably a poster girl on your wall, August Dieter, who was described in a paper in 1906 and 1907. And she was actually 56 years old when she died, August Dieter, the first patient to be described with dementia. And her neuropathology was looked at, and she had high levels of plaques and tangles, which then became the kind of narrative around dementia, that there was a linear deterministic pathway between the neuropathological burden and the manifestation of dementia during life. But this was challenged during the 1980s originally in something called the NUN study, which came out of the United States, which many of you are probably familiar with, which is where 678 nuns from across the United States were recruited by Snowden to an epidemiological cohort. And they were aged between 75 and 104 at baseline. And they agreed to have annual, very detailed assessments of their cognition and to give a kind of medical history and narrative of their lives, and to donate their brains to be examined when they died. And they found that there was not a direct relationship between people who had dementia during life and the amount of pathology in their brains. So there were some people who had who had intact cognition during life, but still had quite a lot of plaques and tangles. And Snowden related this to letters that these women had written when they were aged 21 and entering their vocation. And the relationship was mediated by the vivacity and complexity of the language they used in those letters. So the hypothesis was that the nuns who had had high levels of education during their childhood and advantageous socioeconomic environments, even though they then had a very similar exposures and very little sex and alcohol and all those other sort of risk factors during their lives, the ones who had more reserves in their brains were less likely to have dementia during life, regardless of the Alzheimer's pathology. So you still needed to have some Alzheimer's pathology to have dementia, during your life, but it wasn't a totally linear relationship. And so that inspired some work by Ken Rockwood's group in Dalhousie, and I'd advise you to look up Lindsay Wallace as one of the authors and David Ward, who's now a research fellow in my team. So they looked in a little bit more detail about this by quantifying frailty using a frailty index, which wasn't one of the measures you mentioned, Peggy, but there are many different ways to measure frailty. And so they quantified frailty status and looked at autopsy specimens and found that the frailer you were during life, the less of a burden of plaques and tangles were needed to manifest with dementia during life. And so that was a very kind of definitive relationship that they found. And then David Ward went on to look at longitudinal data, and he published this in JAMA Neurology in November last year, and it got quite a lot of media attention because he showed that 20 years before dementia, those who started to have more frailty detected were a lot more likely to have dementia in old age. So I do want to suggest that frailty isn't a cause of dementia, but it is a mediator. So it changes the threshold at which the pathology manifests with symptoms and signs of dementia during life. And so that is so exciting because that means there is so much we can do at a public health level to change people's thresholds. We're not going to prevent dementia totally, but we're going to delay it if everybody got fitter and more robust and did more exercise and took fewer harmful drugs, which no, I wasn't looking at you. So there are many things that people can do to slow the progression of frailty and to delay its onset. But I don't want to also imply that that is all within the domain of an individual, because I think that frailty is an equity issue. And there are public health, because the people who become frail are more likely to have low levels of education, to have worked in, to have had poorer socioeconomic circumstances, to be disadvantaged, to have chronic disease. All those things are very tied up with frailty during old age. So I think that we need to think about, we need to recalibrate the arguments around dementia prevention in terms of public health initiatives.
Thank you, Ruth. That's very interesting, because we always talk about brain reserve to our patients, and try to explain, why on certain MRI scans, two patients can have the same MRI scan with a lot of brain atrophy, but have very different cognitive performance. And I always say to the patients, because of your cognitive reserve, and that's kind of accumulated through life, and now there's evidence that frailty itself will influence that brain reserve. And everyone wants to have more brain reserve. So, so that's excellent progress. So I'll ask Bonnie to give us some insights on how frailty presents in the acute hospital, and measures that are being done to kind of address it in hospital in the perioperative setting.
Okay, sure. So I guess I work as a geriatrician in Sydney, and I do a mix of acute inpatient work, surgical liaison work, as well as outpatient clinics. So certainly a lot of patients that we see in the inpatient setting, particularly those who are older, are frail. I'd say probably almost all of my inpatients and probably almost all of the patients I see under orthogeriatrics would be frail. I guess in terms of identification of frailty, I don't think it's very well done generally, unfortunately, in the hospital setting. It's not to say that we don't identify patients as being frail, but whether it's actually recorded in the notes for everyone else to be aware of. So certainly as geriatricians, I think we're reasonably good at identifying whether our patients are frail. But sometimes we, particularly for the orthopedic patients, where other clinicians may not be as familiar with frailty. So there's certainly room to move in terms of allowing others other clinicians to be aware of frailty. So certainly, I guess, with the hip fracture registry in Australia, there's now a requirement to document the clinical frailty scale. And certainly at Royal North Shore at the hospital I've worked at, the orthopaedics team are actually becoming a bit better in terms of assessing frailty and identifying patients who are frail and also flagging those patients with us. I guess in the hospital setting, it's important to identify those who are frail because, as Peggy mentioned, and as everyone has mentioned, there are interventions that we can do to reduce or potentially reverse frailty. Because I guess in the inpatient setting, we also see that frailty is modifiable in the sense that patients may be frail at one point during the admission, but they may not be by the time of discharge or vice versa. So I guess in terms of identifying frailty, I guess as well as identifying frailty, I think we also need to implement the interventions which have been shown to have evidence in terms of reversing frailty. So I guess engaging the physiotherapist early on during their admission or getting them to sit up out of bed, ensuring that they are mobilising each day as they're able to. I guess if they are malnourished, incorporating a dietitian, and I guess along with exercise, giving nutritional supplements as required. I guess involving the multidisciplinary team early in the admission, and also performing a medication review. I guess my research largely focuses on polypharmacy or the use of multiple medications, as well as deprescribing, which is the process of reducing or stopping medications that are inappropriate or no longer required. I guess in terms of the relationship between polypharmacy, frailty and dementia, it's quite complicated. And again, I think it's the chicken and egg situation where it's hard to know which one actually came first. I guess when we've looked at studies, there is an association between polypharmacy and frailty and dementia, but it's hard to establish whether there is a causative relationship, just because I guess the frailty scales used are different, and some of the frailty scales also incorporate medication use and multimorbidity into their calculation. So it's a bit of a complicated situation, I think. So it's similar to, I guess, exercise and other aspects where there's a bit of chicken and egg going on.
Sounds like there's certainly room for more research in this area. We've just got a few minutes left now. I just want to ask the audience if they've got any questions for our panelists here.
I have two other quick things to say, sorry, but because we've mentioned the fact that women are more likely to get dementia than men, and women are notoriously more frail than men. This is one of the great paradoxes of aging, that women have a greater burden of frailty compared to their male age-matched peers, and yet they live longer lives. Nobody quite understands that paradox. And the second thing is that we've talked about severe mental illness in relation to dementia. And severe mental illness, particularly schizophrenia, in some research that I'm doing, is a model of accelerated aging and accelerated frailty. So every time somebody has a severe psychotic episode, they seem to deteriorate slightly in functional status. And so that's something that we're looking at, that we're using the prism of frailty to understand dementia and in people with severe mental illness.
Yeah, hi everyone. Thanks for your good contributions to and thoughts about this topic, because I think it's a very relevant and important topic. I have a question for Dr. Bonnie Liu, because I was curious how you think we could engage clinicians like other medical specialists, general nurses in hospitals, in caring well for persons with frailty and dementia. Because I think you as a geriatrician are focused on this topic, but I think it's important more hospital-wide, also in outpatient setting.
Yeah. So I guess in the first instance, I think it's ensuring that the staff within the hospital are aware of frailty. So certainly at Royal North Shore, I've given education sessions to the nursing staff on the orthopaedic wards. I've also given a presentation to the physiotherapists at their education sessions. And I've also given education sessions to the orthopaedics juniors and seniors with each change of rotation. So I think it requires regular education of the staff so that they're actually aware of frailty and actually just to ensure that they're actually able to identify those who are frail in the first instance. And then next it's education on, I guess, the interventions to reduce or potentially reverse frailty in the hospital setting. So I guess education in the first instance, but I guess later down the track, once there is more awareness, and I think there is more awareness compared to 10 years ago in the hospital setting about frailty and its adverse effects, I guess the next steps would be trying to, design interventions which are sustainable in the hospital setting.
Just a very quick comment on that. My own physiotherapist was very interested. I was telling her what we were doing today, and she's asked me to speak to her group of physiotherapists at her hospital because, you know, to do something. Yeah, so that's quite a nice little side thing, I suppose. Yeah.
I guess this is sort of for Ruth. So you mentioned that, you know, women are sort of traditionally maybe more frail than men, but middle-aged women now are really embracing Pilates and yoga and things like that much more than men are. So you think that maybe that will actually translate into some differences later on?
Yes, it's going to be really fascinating, I think, because the women who come to see me in clinic now who are in their 80s and they're worried about cognitive decline, a lot of them left school in their early teens. You know, it's astonishing, and didn't have the ability to work in kind of environments that's made the best of their brain power, and experienced possibly higher levels of depression and anxiety throughout life. And there is interesting studies from Europe. I'm hesitating to talk a lot about these things with Rob in the room, who knows much more about this sort of data, but in Scandinavian countries, for example, which have very advantageous parental schemes for women, there's less of a disparity in terms of frailty in old age. And there are some studies from showing that women who worked in very high pressure industries don't have the same frailty challenges during old age. So I think it will be interesting to see how these cohorts sort of thrive in older age.
Hi, thank you for sharing. Sorry, I'm not too informed on frailty, but I wanted to ask probably specifically more to Andrew. Has peptides been explored in frailty, like BPC and things like that?
In what context, sorry?
And just like helping to reduce muscular atrophy.
So I think in terms of, so if you talk about sarcopenia on its own, so the loss of muscle from midlife onwards probably happens two ways. So gradually you're going to lose your fast twitch fiber, so the muscle that produces that strength and the things that we need to get out of a chair and be really active. So we know that the type 2 fibers, those fast fibers, are the ones that waste, and we think that's due primarily to a resistance or an anabolic resistance with age. So that's your blunting to exercise and protein mainly. So that's a really gradual slope over time. So an older adult probably needs about 3 times as amount of protein per dose than a younger adult. So you don't lose the ability to adapt, but you lose the sensitivity to it. So you have to work harder and take more, but actually it's probably the opposite. We probably take less and don't do as much. So muscle never loses the ability to grow. It's just we forget how to let it grow. The other way of looking at it is that rapid loss in terms of that transition to frailty. I don't think we know a lot about what really drives that quick decline. Unfortunately, I mean, it's inactivity driven. Like most things, it's an insult on top of an aging background that work together, unfortunately. A lot of it is inflammation systemically that impacts that as well. So someone in ICU is going to lose a lot more muscle than just a healthy adult in bed. But it is an inactivity driving that. So great question in terms of what's driving that. I don't think we know. So we need lots of smart people to figure that out.
Thank you.
I have one last question.
Ruth, how are you? Nice to see you
Hello.
again. I noticed that you were careful with choosing your words about frailty being a modifier and not necessarily a risk factor. I was wondering what would be the difference. I know we currently have 14 plus an extra one that came out last year with hearing. Why wouldn't we place frailty as a possible risk factor?
Yes, it's difficult, isn't it? I mean, I think David, I'm quoting David's words here, which is to advocate that frailty is a mediator. So, so it changes your threshold, the threshold at which you become frail. But where you put it on the Lancet Commission is difficult.
All right, so I'd like to thank our panelists for a very interesting discussion.
I'll just stand there. I think I get to close. We'll just leave the panel there awkwardly. Oh, I've pressed the button, sorry. I will close while everyone does the post-event survey except the panel who don't have their phones. So please do the very short survey by scanning the QR code. And while you do that, I will recap the things that I'm now committed to doing as a result of today. First, I will research brain health equity, which seems very important. Haven't done anything about that. I have a growing appreciation of zebrafish. Very cute, very excited. I've written must wear masks during bushfires, important. Sleep 7 hours a night, but not too much. I've also written must make appointment with physio. I have also written, after Jane Thompson's wonderful talk, that I should give my husband a big hug in bed tonight. So thank you, Jane. I would like to particularly thank Tony and Ruth, who travelled from Queensland, and Jane, who's come up from Canberra to be here today. I need to again thank the committee for all the work they've done to get today up, for all of you for coming and staying till the end, particularly Ave, Oksana, and Shannon from the BMC team, particularly Shannon, who actually put the whole thing together almost single-handedly. So the result of the heated argument, which I told you about, is we do have alcohol and other drinks upstairs. So please join us upstairs for more informal chat. And thank you all for coming. That's it, right? I'm done. Okay.